welcome everyone, and a particularly warm welcome actually to the folk from the wider mental health translational research collaboration. We've got, I think, 7 presentations from the mental health TRC colleagues. I invited all the different centres to send someone along, and it's great that you can join us. During the presentations, can you please just switch off your mic and also switch off your video? I can see 1 or 2 colleagues' mugshots moving about. It's slightly distracting, so just switch yourselves off. And then we're going to have the structure of this is to have a certain number of 3-minute thesis presentations, which is led by the students, and there's a competition for these, and we're going to hear some very pithy, quick presentations about what are no doubt much more complex and detailed pieces of work, but it's a great discipline for people to do this. And they're also going to be interspersed with some research showcases. In fact, I'm speaking about a research program I've been working on called RADAR CNS a bit later on. Now, there isn't an opportunity during the 3-minute presentations to have Q&A.
Is now joining.
Okay, there isn't an opportunity to have Q&A for the 3-minute presentations, but there is for the longer showcases. And if you want to just put a question in the chat box, that can be responded to by the speakers. Salah is keeping an eye on that discussion board. Because we don't have time for those questions and answers in the 3-minute thesis section, we actually do have a social sort of networking event in the later on, and an opportunity to work to catch up with people. So I hope that can be made to work. It's a huge challenge. We should be in a lovely auditorium right now, and I think we'd be feeling a buzz of participants as the afternoon was getting underway. It's a big shame we can't be doing that, but on the other hand, I think we can use these platforms very effectively to present our work. Finally, from me, please tweet. There's a hashtag, Maudsley BRC training, so that's in the chat. And I'm going to hand over now and introduce Professor Dave Jones, who is the Dean of the NIHR faculty, so the NIHR Academy, which is responsible really for the whole NIHR training program and much more besides. Dave is a professor of liver immunology. Dave, you're very welcome. I hope you're with us and can share your screen.
Matthew, it's Richard here. In Dave's absence, I've heard him present this presentation a few times, so if he joins us, he can jump in. But perhaps in the interest of time, if I talk through the overview of the Academy, and I suppose welcome on Dave's behalf everyone here. Should we play it that way?
I think that would be best. Yeah. Thanks.
Okay, that's fine. Okay. Hello everybody. I'm not Dave Jones. Wouldn't ever profess to be such an important person. My name's Richard Brown, and I'm currently, for at least a couple more weeks, the training lead for the Maudsley BRC. Dave was planning to give a brief overview and welcome, really talking a little bit about the Academy and how it works for developing new trainees, new healthcare professionals involved in research, really training the future world leaders in areas of translational research, feeding into improving care of patients and those they work with. This is sort of infographic here, just showing the different stakeholders in realising NIHR's mission to improve health and care of the nation through research. Part of that at the bottom is what we're here for today, is the role of the NIHR and its overall remit to attract, train and support the best researchers. And that's throughout their careers here today, we're very much looking at the early career researchers in the hope that they will then join the enormous band of expertise present within the UK, feeding into this NIHR mission. Next slide, Trevor. So over the years since NIHR was formed, the Academy, which is all people who are supported in their training by NIHR in one way or another, and that includes the BRCs, has grown enormously. So it counts now to over 4,000 individuals. So everyone here today who is supported by NIHR in one way or another is a member of that academy. And the largest proportion of those are people who are funded through one of the training programs or through the infrastructure such as the BRCs. Next slide. This just shows a breakdown of the different award categories, and we'll jump on. I think we're in the green section there, which is NIHR infrastructure and schools, including the BRCs. Next slide, Trevor. And this is just an indication of the way in which NIHR arranges its support in different strands, both the personal awards, people applying for individual fellowships, people who are supported via the various integrated training pathways, including doctors, nurses, and AHPs. And the final arm is the infrastructure and capacity building structures of the research councils, the ARCs, and the patient safety and translation research centres. Next slide. I think most people who are supported by the BRC are aware of the pathway going through pre-doctoral fellowships, through doctoral, advanced, and ultimately up to research professorships. So there's a wide range of different funding support there for people at different stages of their research careers. All of this information is obviously widely available on the NIHR website, so I won't spend any more time describing this in any detail.
Louise Vale
is now exiting.
Okay. I think I'm going to jump over this as well, because this is some of the technical detail of the funding schemes. It's not really our business today, but again, I refer everyone to the NIHR websites. So anyone who's approaching the end of their PhD, who may be looking for applying for further NIHR funding, all the information that you need will be on the fellowship pages of the NIHR. Next slide. And again, advanced fellowships would be the next stage, available through the NIHR for those people coming off of PhD studentships supported by the BRCs, but also students coming, wanting to continue research in areas of applied and translational health and mental health, coming from different funding pathways or supported by other funders for their PhD. And move on. And another option for those finishing their PhDs who want to maybe develop some skills in a different area, there's also another pathway, short-term funding, allowing people to develop skills and enhance their expertise in an area outside of their PhD. Usually it's a stepping stone to an advanced fellowship. Next slide. The final, I think, is one of the final slides. The NIHR has, in its recent review, which led to the formation of the Academy, set out a range of different strategic themes, and these aren't prescriptive and they're not exclusive, but these are areas in which NIHR is particularly keen to develop expertise and build capacity. And it's very reassuring to see that mental health is one of those themes. But also, I think mental health intersects very, very strongly with the other research strategic themes of social care, public health, and particularly health data science. And we'll be hearing more about these areas, I suspect, in all of the 3-minute thesis presentations, but also in the showcase presentations that we'll come to. The other point just to mention about NIHR's focus going forward is a real aim to develop methodologists or researchers with specific methodological expertise. And again, there's increasing emphasis on providing training support for those individuals, not just within the context of their PhD, but afterwards and in the transition to the next stage of their research careers. Next slide. Just to say, the NIHR works very closely in partnership with other healthcare charities, and in the last couple of years have set up a number of joint funded fellowships with these research charities, and the number is growing year on year. Next slide. And I think I'll skip over this. This is just some additional opportunities which are available to current Academy members. So I've just lost my screen. Yep, that's fine. Jump on. And I think very much near the end, just to remind all of the PhD students currently with us, is the fantastic opportunity afforded by NIHR in terms of the research training camp. This is an annual event, extremely popular and extremely valuable experience for all those who have attended. Indeed, it's been held in very nice surroundings, giving a chance for everyone to get together. I am not aware of what the plans are for the next training camp. It would be a great shame if it is virtual, but would nevertheless, I think, be extremely rewarding for everyone who was able to take part. Jump on. And I think I'll jump on past this because I really don't know what he was going to talk about. Moving on, and that is it. So that is the email address if anyone wants to contact the Academy, but all the information about who to contact about different topics is available on the website. Okay, and that's the end of Dave's presentation. I'm sure I didn't do it justice. Dave, are you there?
Here.
Ah.
I was thinking, Richard, I was thinking that we're going to have the same presentation until I realised you were giving my presentation.
You weren't here, Dave, and we were on a very, very tight schedule. So I said, I'm going to pretend to be you, but I would not do the job that you would have done. I might have done it quicker. If you do have any words of wisdom to share with us, I'm going to go on mute now for a second.
Yeah, so Richard's very modest, and he is a absolute champion in NIHR and hugely valued. Yeah, so my name's Dave Jones. I'm the Dean in the NIHR Academy. I'm also a working hepatologist, and that's where I've been, so I apologise for being a little bit late. So my main comment is to say welcome to all of you. I see there are over 80 people on the call, which is absolutely brilliant, because you are all an important part of the NIHR Academy family, and the NIHR Academy was set up to continue with a lot of the very good work we do on training, but to value people ever more. And Richard just touched at the end about some of the things that we offer people around the training camp, and the awards that can take you to another centre to gain different experience. The wraparound training around leadership and skills, and all of that is to help you become the best researchers that you can be. So we support people to be successful in their careers, not just to do a piece of research. So clearly your research is the main focus, but it's around skills to be successful and to be hugely, valued. Just a couple of general observations. I'm particularly keen to talk to you because mental health is absolutely a massive priority for NIHR, quite rightly. In a number of different ways, it's a priority in its own right because of the impacts on the population. It also features in our drive around multimorbidity, multiple long-term conditions, as we tend to talk about these days, because of the issues around physical health challenges in people with mental health problems and vice versa. So it is a priority area in lots of different ways. Now, that isn't just empty words. It means that for fellowships and so on, there is a prioritization in this area. So it really is a good time to be thinking of working in these areas. And I'm guessing most of you will be at doctoral level, which is great. The infrastructure provides fantastic support for people doing PhDs. But the expectation and hope is that you will continue with your academic careers. And the main thing I wanted to do was to just sell the NIHR Advanced Fellowship to you. This is all our postdoctoral offerings rolled together. It has been changed substantially to take account of people's needs and career differences. Lots of flexibility to allow people with different career tracks and different caring needs and needs for whole time or less than full time working, and the speed at which people want to do research. And so we've made it as friendly as we possibly can for people. We can support people to do in practice work, so clinical work for all professional groups during their advanced fellowship. So those people perhaps from a nursing perspective, psychology, so on and so forth, we will support people to do their clinical practice as part of their fellowship. So clinical academia, which used to be for medics only, for historic reasons, is now open to everybody. And we're just extending that now into social care and into public health. So very exciting opportunities to do all of that. And of course, the fact that it's a strategic theme means that there is an uplift in application. So if you've ever thought of following careers through into a postdoctoral fellowship, the offering, I think, for people working in mental health from NIHR is exceptional at the moment. I don't see actually that changing anytime soon. For those of you, there will be some people I suspect at master's level. All of the above also apply to our doctoral offerings. So please don't be shy at applying for personal awards, as well, of course, as the awards through the BRC. So as Richard will have told you, I guess, I started my life in NIHR looking after infrastructure training. It's very dear to my heart. The numbers of people we train are fantastic. The outputs are fantastic as a group. But please, please, please stick with what you're doing. It is seen as being important. It's incredibly highly valued. I absolutely must emphasize that. I think we've made the fellowships to follow on attractive for people. So think about those. And then most of all, you have a sort of wonderful time in mental health because we absolutely see it as a priority area. So please think of us for your fellowships as you move forward. And I've almost certainly destroyed your very tight timing with that. So apologies for being slightly late.
But, very much appreciate you making the time. So I'm going to hand back to Richard for the 3-minute thesis presentations.
In a moment, we are going to be kicking off with the very first presentation from Sophie Forson. Okay, Sophie, so I've got to say, are you ready?
I am, yes.
Okay, so
in the UK, breast cancer affects 55,000 new women each year. Detection and treatment improvements mean survival rates have doubled in the last 40 years. However, even survivors, so those who have been treated with curative primary treatment, still experience ongoing physical and psychological challenges. Distress is prevalent in these women, with estimates of 15 to 40% experiencing distress. The period after primary treatment is important as patients can feel uncertain due to medical contact ending, individuals are working towards getting back to their pre-cancer lives, and some have fears around recurrence. Distress has also been found to be associated with increased healthcare use and poorer outcomes. These women also experience physical symptoms, particularly those receiving ongoing hormone therapy for recurrence risk reduction. Hormone therapy is prescribed to estrogen receptor positive breast cancer, which makes up to 70% of all breast cancers. Side effects of this treatment include joint pain, menopausal symptoms, and fatigue, and can be particularly problematic. These symptoms are often associated with distress. So, what do we know? Distress is prevalent, physical symptoms are common and can be difficult, so what can be done about it? That's where my PhD comes in. The wider aim is to help women with breast cancer manage their distress and physical symptoms. To address this, I'm conducting several different studies informed by health psychological theoretical models of distress and previous research to develop an appropriate evidence-based intervention. Using a mixed methods approach, the studies will explore psychological factors based on cognitive behavioural models, so focusing on the relationship between thoughts, behaviours and emotions, and in therapy how to disrupt these unhelpful cycles, and more third-wave approaches such as the psychological flexibility model used in acceptance and commitment therapy, which aims for a willingness to experience thoughts rather than trying to change them whilst pursuing meaningful valued life. There are studies focusing on cognitive behavioural elements in cancer, so this PhD will focus more on this gap in the literature by exploring the psychological flexibility factors. A systematic review will determine the evidence base for these psychological flexibility components in cancer patients, whilst a qualitative study will explore more of an understanding of acceptance in these factors, what these mean for breast cancer patients, and how patients feel they manage their distress and symptoms. Longitudinal studies will determine how cognitive, behavioural and psychological flexibility factors are associated with distress and experience of physical symptoms, whether they predict later distress, and whether they mediate the relationship between symptoms and distress. These studies together will inform the adaptation of an existing mobile app-based intervention. The results may indicate that targeting management of symptoms, so creating a different way to have a relationship with thoughts around this area, could then reduce distress. It may be that an integrated model is developed, informing the self-management app-based intervention. Patients will be involved throughout and take a key role in the intervention development. So to summarise, the studies of this PhD will help build a model of distress and managing symptoms to develop an intervention to help women with breast cancer on hormonal therapy.
Moving on now, so Florian Hansen will be joining us from the Department of Psychology here at King's. Okay, so Florian, are you ready to start?
Yep.
Okay, so the slide will progress.
Schizophrenia is a diagnosis associated with severe disability. It is most commonly characterized by the positive symptoms associated with it, namely hallucinations, such as hearing voices no one else can hear, and or delusions, distressing beliefs that are unrealistic and in contrast to societal norms. The contemporary negative symptoms are lack of motivation or goal-directed behavior, being unable to see joy happening in one's future, lack of interest in social relationships, poverty of speech, may be an amount of words or concrete content, and or reduction in emotional expression through the face, speech, intonation, and or gestures. As you might already suspect, it is these symptoms that are often leading to disability. Schizophrenia is very expensive. In England alone, costs are estimated at 18 billion a year, which equates roughly to 350 million a week, a figure most of us might be familiar with through another context. 3/4 of those costs are due to productivity loss, which is mostly credited to negative symptoms. While positive symptoms can be successfully treated or at least controlled, negative symptoms remain barely treatable, if at all, while research into it is advancing very slowly. A reason for this might be that the available measurements of negative symptoms are at best lengthy, costly, and still have variance problems associated with them. They are also computerized measures, but they are not very popular or engaging. Don't get me wrong, some modern tests are quite good at measuring negative symptoms, but none is feasible for mass implementation across the NHS, maybe due to inaccuracies, costs, or the willingness of service users to engage with them. In contrast, an optimal measure would be relatively inexpensive, and it be at least semi-automated and therefore relatively objective. Moreover, the measuring process itself should have ecological validity, meaning it relates to how symptoms occur. Finally, service users should like the measure and want to engage with it. And here's where we get to virtual reality. The technology is very popular among service users and has become cheap enough for the mass market. VR at room scale, meaning you can move through it and interact with it, is really hard to explain. It is an entire experience from a visual, auditory, and partially tactile sense. What you need to take home is that VR allows us to create lifelike but completely controlled simulated 3D environments. This enables measuring of complex behaviors in response to complex stimuli, or in our case, the absence thereof. My research takes validated experimental paradigms to measure negative symptoms and adapts them for virtual reality. Service users will then be asked to engage in activities similar to everyday life while their behavior is recorded. After a prototype was developed, service users' feedback will be used to improve the measure and make it more acceptable. Then the final version will be tested by more people, including healthy controls, and their scores will be compared against traditional measures to ensure we are actually measuring negative symptoms. A VR measure of negative symptoms associated with schizophrenia holds the potential to be more accurate than traditional measures, while also being more affordable and more popular among service users. If so, it might drastically accelerate research, specifically treatment development, which in conclusion might actually make more money available to the NHS. Thank you.
Okay, so moving on. Timothy, are you with us?
Hi, yeah, can you hear me?
I can hear you. Are you ready to start?
Yeah, I am.
Okay, so off we go.
So you are your brain. Everything you think, experience, and perceive is a result of some complex emergent process arising from the 1.5 kilo of lump inside your skull. It is often said that we see the world differently. Pain is similarly diverse. It's a unique experience and it's shaped by each person's prior experiences and genetics. Pain is in your brain, and as such, what affects your brain can affect pain. Being cut with a scalpel hurts a lot less while you're under anesthesia. A toothache is unbearable when lying awake at night, but barely noticeable when you're chatting with friends. When you're scared for your life, you can run on a broken ankle. Drugs, context, and cognition all work through changing the way your brain processes pain. Neurodegeneration, which can result from common disorders like Alzheimer's and Parkinson's disease, profoundly impacts the normal functioning of the brain. This includes these same neural circuits that give rise to our conscious experience of pain. This is important because we tend to think about Alzheimer's as a disease affecting memory and Parkinson's as a disease affecting movement, but many of these patients also suffer from severe chronic pain, and this has a massive impact on their lives. Patients with Parkinson's often rank pain as one of their most troublesome symptoms, and in patients with Alzheimer's disease, difficulty contextualizing and expressing pain can be misattributed to behavioral or psychiatric issues. So at this point, you may be wondering why these patients suffer from chronic pain. It seems this is partly due to the way these pain circuits are affected by neurodegenerative pathophysiology in the brains of these patients. This results in pain being processed and experienced differently. For example, when you evoke pain in these patients in a lab setting, they experience a heightened level of pain compared to healthy individuals, given the same stimulus intensity. This not only has implications for understanding the development of chronic pain in these patients, but also for treatment. Some of the drugs we use to treat pain act by working on these same brain circuits that are affected in these patients. This may mean they do not work the same in these patients as in the general population, but no one's really tested this idea yet. So my PhD project aims to use functional neuroimaging to look at how these patients' brains are actually processing pain and to test whether giving them a pain-relieving opioid drug produces different effects on their brains. Currently, there are no treatment guidelines for pain in either of these disorders, despite this being a huge clinically unmet need, and I hope that research like mine will help open the door for evidence-based treatment of pain for these patients. Thank you very much for listening.
Okay, so moving on. So we have Jessica Mundy from the SGDP Centre here at King's College London. Jessica, are you with us?
Hi, yeah, I'm here. My internet's a little bit today, so let me know if you can't hear me.
Yes, you are a bit breaking up. We're going to do our very, very best to pay attention. Yeah, I can hear you, so you're a bit fragmented. The audience and the judges. Okay, if you want to pause a second. So if you're ready to begin, are you ready? Hi, Jessica, can you hear?
Yeah, I'm ready. Can you hear me, Richard? Major disorder and bipolar disorder are grouped together as mood disorders. Major depressive disorder is classified by persistent low mood and ability to experience usual joy and pleasure in daily activities. Bipolar disorder is divided into two types, type 1 and type 2. Type 1 is characterized by the presence of something known as mania. Lots of the people mistakenly associate mania with intense happiness, but mania is actually very different to this and can involve symptoms such as inflated self-esteem, racing thoughts. Somebody experiencing mania might engage in reckless or impulsive behavior, such as spending lots of money in one go. This is why mania is much more problematic than simply feeling extremely. So mania is present in bipolar type 1, but what about type 2? In type 2, something known as hypermania, below in Latin. So hypermania refers to a less intense form of mania. Hearing this, you might wonder why hypermania is considered a problem. Well, in bipolar type 2, the individual also experiences depressive episodes from a depressive to hypomanic state, which is extremely. Mood disorders bring higher rates of associated diseases such as cardiovascular disease. They are also associated with high levels of mortality, which means death rate, due to the proportion of affected individuals who die by suicide. Therefore, understanding the causes of mood disorders is crucial for developing effective treatments, which will hopefully profoundly improve people's lives. Risk for developing mood disorders is inherent. No single gene exists which causes mood disorders. Rather, a combination of hundreds. You can think of these genetic variants as tiny puzzle pieces that make up your entire DNA sequence. Since there are hundreds of thousands of these puzzle pieces, the journey towards understanding the genetic basis for mood disorders is rather complicated. However, studying pieces are found in the DNA affected and traced up. This is the main aim of my thesis. My research will involve participants who have signed up to the Genetic Links to Anxiety and Depression study and the UK Bio. Participants in both studies have provided lots of information through answering online questionnaires about their past and current psychiatric symptoms. They also provided a saliva sample. This will allow me to specifically study participants who have experienced depressive disorder by analysing the DNA from their saliva sample.
Okay. So our next presenter is Suzanne O'Brien from the Department of Forensic and Neurodevelopmental Science here at King's. Suzanne, are you with us?
Yes, I'm here.
Very good. Are you ready to kick off and start?
Yes, I'm ready.
Okay, so if you start as soon as your slide appears.
Okay, so the title of my project is called Born Bad, Can We Treat Children at Risk of Becoming Psychopaths? So conduct problems are characterized by repetitive and persistent patterns of antisocial behavior in children. And there are several factors that modulate the severity of conduct problems, and one being the presence of callous unemotional traits, or CU traits for short. So those with high levels of CU traits, for example, will exhibit more severe conduct problems, lack of empathy, lack of guilt, commit more violent crimes at a younger age, etc. And it's this subgroup of children that are thought to be at an increased risk of developing psychopathy in adulthood. And we also know that children with conduct problems have abnormalities in brain regions such as the amygdala and in the ventromedial prefrontal cortex. Currently, the only treatment available for children with conduct problems is group parenting programs, and the gold standard parenting program in the UK is known as the Incredible Years. And during this 14-week program, parents will learn new parenting strategies in order to help manage their child's behavior. However, nobody has examined whether these behavioral changes that occur as a result of the parenting program are associated with brain changes. Therefore, in study 1, 80 boys between the ages of 5 to 10 underwent MRI scans and assessments of antisocial behavior before and after their parents took part in the Incredible Years parenting intervention. The aim is to determine if differences in brain anatomy and function can actually change in parallel with reduced antisocial behavior, and also to determine if brain differences can actually predict behavioral alteration. However, there is about 40% of children that do not respond to parenting programs, and it's mainly these kids who have the high levels of callous unemotional traits. Therefore, there is a critical unmet need for research to develop new effective treatments for this subgroup of children, and that's where study 2 comes in. So recent research has also shown that children with high levels of CU traits, they have reduced activity in their oxytocin system compared to those without CU traits. And this is important to know because oxytocin does have diverse positive effects on prosocial behavior. Therefore, the aim of study 2 is to conduct a pilot fMRI study to test the hypothesis that compared to a placebo, a single dose of intranasal oxytocin can temporarily shift abnormal neural processing towards normal in a group of high CU boys versus low CU boys, all of who previously did not respond to the parenting program. And if we find evidence that oxytocin can regulate abnormal neural processing in high CU children, then it could form the basis of research into the first therapeutic intervention for this subgroup of kids who there is currently no treatment for. Thank you.
Okay, thank you very much. So moving on, Abigail Tutquil.
Hi,
Abigail. Hello there. Are you ready to start?
Yeah.
Okay, so Abigail is joining us from the SGDP as well, King's College London.
Anxiety and mood disorders are common, debilitating, and difficult to treat. Anxiety disorders are characterized by excessive fear and anxiety and impairing features such as avoidance behaviors, physical anxiety reactions, and panic attacks. Clusters of symptoms differentiate one anxiety disorder from the other, including general anxiety disorder, panic disorder, agoraphobia, specific phobia, and social phobia. The mood disorder spectrum includes depression, characterized by persistent sadness and the reduced ability to experience pleasure. It's often experienced for extensive periods, becoming either chronic or recurrent, and contributes to a large proportion of suicide. Depressive episodes are also prevalent in rarer mood disorders, notably bipolar disorder, which also presents with periods of abnormal elevated mood, which is more severe in bipolar type 1 than bipolar type 2. These disorders often overlap, with many people experiencing different types of both anxiety and mood disorders. Responses to existing treatments are varied and prescribed in a trial and error fashion. To improve the prevention and treatment of these disorders, a better understanding of their risk factors is needed. Decades of research has shown that the risk of having anxiety and mood disorders is partly genetically influenced. We now know that it's not a small number of genes that contribute to this risk, but hundreds and thousands of variations of genes that increase the risk by a small effect. To find these thousands of risk genes, very large sample sizes are needed. My thesis will use the Genetics Links to Anxiety and Depression study, the largest study of anxiety and depression, to address 4 questions. To what extent do the genetics of anxiety and mood disorder subtypes differ? If so, which genes are different? What do these genes do? Could these genes explain why anxiety and mood disorder subtypes differ in their symptoms? Although genetics play an important part in the development of anxiety and mood disorders, the environment is also an important factor. Severely stressful traumatic experiences are well-established risk factors for anxiety and mood disorders. Although primarily recognized as environmental exposure, self-reporting trauma is partly genetically influenced. This may reflect genetic factors influencing exposure to environmental stresses. However, it may also reflect the genetics of traits that make someone perceive and remember an event as stressful, and the willingness to report potentially stigmatizing events. My thesis explores the behavioral traits that contribute to the genetic component of self-reporting trauma. Only a portion of those who report trauma have an anxiety or mood disorder. Some people may carry a greater genetic risk to respond to the negative effects of traumatic experiences. This interaction between the environment and genetic vulnerability may explain why those with a higher genetic risk to anxiety and mood disorders will present with the symptoms when exposed to trauma. My thesis will aim to identify the thousands of genes that interact with traumatic experiences that increase the risk of developing anxiety and mood disorders. Thank you.
Okay, moving on. So next we have Lizzie Tudor. Lizzie, are you with us?
Yes, I am.
Okay, are you ready to start when the slide progresses?
Yes.
Yeah, okay, so Lizzie is joining us from the Department of Health Services and Population Research.
Individuals with severe mental illness, such as schizophrenia, are at a twofold risk of developing diabetes and experience a reduced life expectancy by approximately 10 to 15 years compared to the general population. People with severe mental illness and type 2 diabetes are also less likely to receive essential healthcare that could help to reduce preventable diabetes complications such as stroke, lower limb amputation, and blindness. This population are also more likely to experience financial poverty, low educational levels, and social stigma, which act as barriers to access services to prevent and improve illness. These health inequalities are ethical and economic issues that must be addressed. There are a limited number of integrated non-pharmacological interventions that have been designed to improve both type 2 diabetes and severe mental illness, and they have had varying degrees of success in improving diabetes-related and mental health outcomes. A lack of integrated physical and mental health care and interventions adds to this problem. My PhD aims to address this. Understanding service user and staff experiences are key to improve the weaknesses and enhance the strengths of healthcare. My study will use the experience-based co-design approach, which involves bringing together service users, carers, and staff in a series of events using a 6-stage cycle. Observations of daily practice at the healthcare service and video and/or audio recorded individual interviews of service users, carers, and staff will be conducted. Service users and carers interviews will be edited into a short film that highlights touch points, which are positive or negative emotions throughout participants' care experience. Participants view the trigger film and power imbalances between service users and staff are shifted by encouraging participants to reflect on the touch points identified. Participants then agree on the priority areas of improvement and then form co-design teams to develop the interventions to improve the challenges identified. A celebration event is held to review and share the success of the participants' work, which is also shared with wider members of the Trust. My PhD aims to assess if it is feasible, acceptable, and effective to use the experience-based co-design approach to implement an intervention to improve the care of people with type 2 diabetes and severe mental health illness. If successfully applied, the experience-based co-design approach could provide a genuine voice for this population who disproportionately experience health inequalities and encourage a positive shift to improve both service user experience and staff well-being. Thank you.
Okay, I didn't have a chance to say thank you to Lizzie for her presentation. I'll do that now. So moving on, I'm very happy to welcome our first speaker from outside of King's College London, Dr. Nathan Hineker, who's joining us from the University of Southampton. So Nathan, are you with us?
I'm here, yep.
Okay, are you ready to go when the slide progresses?
Yep, should be.
Okay, so over to you.
Anxiety disorders are the most common psychiatric disorder, affecting roughly 1 in 10 people. Although we have treatments for anxiety disorders, they're not ideal, as only about half of patients get better with first-line treatments, and the medications that we use can cause unwanted side effects. We need to develop better treatments to improve the effectiveness of them and to reduce side effects. It's necessary to test a new treatment in placebo-controlled trials, but approximately 30% of patients in psychotropic drug trials show a clinically significant improvement when treated with a placebo. This can reduce the chance of detecting the effects of the drug. In anxiety, improvement following a placebo is known as placebo anxiolysis. Despite its magnitude and importance, placebo anxiolysis has been largely unexplored. Placebo responses have been explored in other domains, and placebo treatment is known to activate neurobiological symptoms that control incoming sensory information. This has probably been best demonstrated in pain, where placebo analgesia is known to involve the endogenous opioid system. Although placebo anxiolysis has had little exploration, there is some limited evidence to suggest that control of emotional state induced by a placebo might also involve the endogenous opioid system, and that this system might be important in regulating anxiety. For this PhD project, I've got 3 aims. First, I aim to develop a novel, clinically relevant paradigm to induce placebo anxiolysis in healthy volunteers, as such a paradigm currently does not exist. I plan to use the 7.5% CO2 inhalational model of anxiety, which mimics the features of generalized anxiety and shows response to standard treatments. The stimulus, in this case the gas, can also be easily manipulated to convince the participants that a treatment is helping them to feel less anxious. This project is ongoing. Second, I plan to use this paradigm to test the hypothesis that placebo anxiolysis is mediated by the endogenous opioid system. Finally, I aim to understand whether endogenous opioids are important in regulating anxiety outside of a placebo context by scanning healthy volunteers' brains as they view threatening images and either reappraise or react to these with and without blockade of the opioid system. The endogenous opioid system might emerge as a potential therapeutic target for anxiety disorders. Thank you.
Okay, Becky Green. Becky is joining us from the Department of Basic and Clinical Neurosciences, King's College London. Becky, are you with us?
Good afternoon, yes I am.
Okay, and are you ready to start when the slide progresses?
I am indeed.
Okay, so over to you.
So first of all, a bit of background. So dementia is a global health concern, and we know that genetic and life course risk factors such as low education and diet play a role in its development, and also that there exists a period before diagnosis where disease mechanisms are accumulating, providing a great opportunity for potential intervention. However, little is known about this period, as identifying individuals in this phase is challenging. Using studies that have followed individuals over a long time, we can identify early indicators such as biological markers preceding diagnosis, an example of which may be metabolites. So metabolites such as fatty acids are the products of biological events and are influenced by both genetic and environmental factors, providing a holistic view into the health status of an individual. We aim to investigate whether metabolites could be valuable markers of early changes relevant to dementia and evaluate whether these associations are influenced by life course factors. This work could help reveal early disease mechanisms, identify those at risk, and guide interventions. So moving on to our research methods on the right here, to do this we use the 1946 British Birth Cohort Study, which is a large, well-characterised cohort with information across the life course. We then measured all metabolites able to be captured, so roughly 1,000 metabolites, in 1,800 participants at age 60 to 64, and looked at whether they were associated with several measures relevant to early dementia, such as brain scans and cognitive tests measured at the same time point, as well as 5 to 9 years later. To capture the effects of metabolites and the relationships between them, we used multiple analytical designs to identify associations of single metabolites, metabolite pathways, and groups of similarly expressed metabolites. Integrating these, we zoomed in on those that are likely to be key drivers in these pathways and groups and looked at biological functions to understand potential underlying mechanisms. So for step 2, once we identified these metabolites and metabolite groups, we explored whether any associations were sensitive to life course factors. In this regard, we identified some metabolites, such as a particular group of lipids, that were entirely explained by early life factors, suggesting a potential mechanism by which these influences may embed biologically. We also identified a group that were independent of known risk factors and honed in on an influential metabolite called palmitoylcarnitine, a fatty acid relevant to impairments in energy production. We will next seek to understand whether these relationships are in the causal pathway using statistical genetics techniques. If this is the case, then they may be useful for diagnosis and intervention. And casting your eyes to step 3, we wish to establish whether our findings are able to accurately predict individuals that go on to experience dementia, which will further reveal whether they may show utility as markers of early dementia. Thank you.
We're moving on now to Danielle Huisman for the final presentation in this first session. So Danielle is joining us from the Department of Psychology, the Health Psychology section. Danielle, are you with us?
Yes, I am.
Okay, and are you ready to start when the slide moves on?
I am.
Okay, so over to you.
My thesis explores biopsychosocial mechanisms of abdominal pain, both in inflammatory bowel disease, IBD for short, and irritable bowel syndrome, IBS for short. IBD is the group name for Crohn's disease and ulcerative colitis. It is an autoimmune disease characterized by inflammation of the gastrointestinal tract, and as such has a clear biological cause. Its dominant symptom is abdominal pain. People with IBD are likely to have periods of inactive disease, but they may not experience relief from their abdominal pain during these periods. That will be the focus of my thesis, both because abdominal pain impairs quality of life in IBD and because abdominal pain that isn't associated with inflammation poses a clinical challenge. We need to learn how to manage it better. I explore if ongoing abdominal pain in IBD may be explained by mechanisms thought to underlie IBS. There seems to be an overlap between the two conditions, as you can see on my slide, and chronic abdominal pain in IBD is frequently classified as IBS. Just like with IBD, people with IBS struggle with abdominal pain. However, the disorder is considered to be medically unexplained, meaning that there seems to be no underlying disease causing it. Instead, IBS is commonly explained by dysregulations in pain processing. Pain isn't experienced at the location where tissue damage occurs. That's only the trigger. Information on what is happening locally is first transduced and then transmitted to the brain, where it's perceived and interpreted. The brain gives us information about the location, modality, and intensity of the brain, as well as the discomfort the pain poses. The brain not only receives information, but also signals back to lower areas involved in pain perception. This feedback serves to impact transmission, to modulate the pain, usually to tone it down. In IBS, this feedback process is thought to be impaired. Furthermore, it is believed that brain areas involved in processing emotions and cognitions influence these feedback pathways, which is why psychological factors such as anxiety and depression may influence pain perception, and hence we call it the biopsychosocial model. To investigate the relevance of the model in IBD, I carry out experiments assessing different aspects of pain processing and explore associations with psychological factors. Finally, I assess support for the model by asking people with IBD how they make sense of abdominal pain during an active disease and how they see IBS in relation to their IBD. Thank you.
So that brings us to the end of the first session. I'd like to thank all the early stage PhD students who presented to us, and congratulations to all of you for staying within the 3 minutes. I'm moving on now. I'm going to hand over to Professor Matthew Hotoff and his members of his team, who are going to present the first of the research showcases. So Matthew, over to you.
Thanks very much, and thanks everyone who's presented so far. I thought that was fascinating and really difficult to score, actually. Really enjoyed it. So we're going to talk now about a program I've been running called RADAR CNS. This is work in progress, so you're going to hear quite a lot about how we've set it all up and not very much about results. And it's going to be a bit of a relay between myself and a few colleagues there. Now, I actually have to, unfortunately, Sarah Simlett and Faith Matcham aren't able to make it for various reasons. So Faith's I think on the call, but suffering with laryngitis and simply can't project her voice. So you're going to have a run of a bit of a mangle that I apologise for, but it wasn't intended this way. So I'm just going to hope my slides work. There we are. That's working. Okay, so I'm going to introduce RADAR CNS broadly. Then I'm going to hand over to Amos, who's going to talk a little bit about some of the sort of tech developments. And then there's going to be two sections, which I'm afraid you're going to have to put up with me presenting on the patient and public involvement and a study of depression before handing finally to Nick Cummins, who's going to talk about analytics strategies. So, and just to say, this is a private-public partnership. It's a pre-competitive funding body called Innovative Medicines Initiative, which is a European funding body. These studies are run in conjunction between the public provider, which is the European Commission, and a number of pharmaceutical companies, and you can see those there. So I'm just disclosing that this is work done with pharmaceutical company support as a pre-competitive consortium. The point about IMI is that it's about trying to crack a really big problem and bringing together industry, academia, small and medium-sized enterprises, and so on, to all address that together. So if we think about it, the world changed not that long ago to an almost unrecognizable, now my display is going off, so I'm hoping that you're still hearing me. If we could just go, I don't think I can go back. Okay, so everyone carries a smartphone in their pocket, and that's a complete change in the last 15 years or so. And these smartphones are hugely powerful, and there is an enormous interest in health about trying to develop mobile health technologies. And you'll see infographics like this on the internet making huge claims about the way in which the sensors on your smartphone and the ability of your smartphone to provide interventions can really change the way in which we practice medicine. And what's attractive, particularly in mental disorders, and I guess the broader CNS disorders, is that you can imagine a spectrum of a disorder from people at risk but healthy, developing a prodrome, to a diagnosed disorder and long-term disease management, where at the moment our data really tends to be intermittent, sparse, and lots missing. There are various ways in which one can use web-based platforms and so on to increase the frequency of that data. But what we really want to get to is something which is continuous, pervasive, and passive. So if you think about being able to get data from a smartphone, you can actually do that without the person having to do a great deal. So what sort of data are we talking about? We're talking about a whole range of different types of data. And when I say smartphone, I also mean wearable devices as well. So one can track, for example, seizure detection using certain wearable devices. We can look at cognition both actively and passively on smartphones. Using a wearable device, you can get heart rate and even heart rate variation. You can get digital usage, the app usage on the phone battery, measures of gait and the step count, geolocation, sleep data, environmental data, data on sociability, and voice data, as well as mood, which might be got from questionnaires. So you can actually get a readout of many of the higher functions of the CNS simply from a smartphone and a wearable device. Now, obviously, these aren't the same as the quality you might get from a sleep lab, but they do have the advantage of a very large amount of continuous data with some ecological validity, which you wouldn't have from other technologies. And the purpose of this is that I'm just going to advance this animation. If you think about a disorder like depression, you might have someone who is having symptoms, relatively few symptoms here, but is at risk of a relapse. They reach a certain point where they have a relapse, and you would like to know about that relapse in advance. And the purpose of RADAR CNS, one of the purposes of RADAR CNS, is to see whether these data streams, for example, sleep or motor activity, heart rate variability or whatever, could actually help one predict a relapse of depression in the future. Put formally, we're trying to transform patient care through remote assessment using wearable technologies, and we're trying to look at characteristic patterns or biosignatures in data that can predict relapse or deteriorations. We're actually looking at 3 different conditions: depression, MS, and epilepsy, and the purpose of this is to create a pipeline for developing and testing remote measurement technologies to build an infrastructure which can be applicable to other disorders, and Amos is going to talk more about that soon. It's an international consortium with academic and industry partners, and there's a very strong patient advisory board, which I'll come on to. And we have organized ourselves with this program into 3 main research clusters, one focusing on the different clinical disorders, MS, epilepsy, and depression, a second looking at the technical challenges, which is building a platform and the analytical technologies which go to that, and the third looking at translational pathways, which is how this would actually be implemented in real life, where we have a lot of engagement of both patients and also clinicians trying to help us develop this. So I'm going to hand over now to Amos Follerin, who's going to talk about the RADAR base platform. I'm stopping my control, Amos, and you need to pick it up now.
Hi, Matthew. Thanks very much. I've just put a few slides together to give a kind of overview of the platform that sort of was that kind of came out of the RADAR CNS project. There's a little bit more about this if you're interested on the links on this slide. To support the data collection in RADAR CNS, we were tasked with a technical work package looking at how exactly we would be able to kind of collect this kind of data. And if we kind of roll the clock back, a best part of 4 years now ago, the landscape was quite different even to what it is now in terms of smartphone ownership, wearable devices on the market, and so on and so forth. But there was definitely the capability to collect data with smartphones. We had kind of access to the sensors on the phones themselves. So even within a kind of modest range smartphone, you've got a whole sort of suite of various types of sensors that perform kind of normal functions in the phone, whether it's a gyroscope that detects what the orientation of the phone is, or a magnetic sensor that shows you which orientation the phone is pointing, with respect to north-south. So these are all kind of parts of the phone, but we can actually access the data that's collected through these. And similarly, there are various wearable devices that we have on that we can measure a number of physiological parameters with, and these set of physiological parameters are kind of growing month on month. We get new sensors, sort of modalities arriving in the market. So common things that we're doing in CNS is collecting things like heart rate with various devices, I should say. So the things that support epilepsy are quite different to what we're using in depression. So we're measuring heart rate, we're measuring things like electrodermal activity, temperature, step counts, etc. There is also another domain that is a little bit separate to RADAR CNS that we've been working on more recently, and this is around incorporation of IoT type sensors. So these are things we might put on inside of a room to look at motion detection or air quality or UV, or there are literally hundreds of different kinds of sensors in this area. We can now collect that data and stream it into the same platform. And then finally, there are those really represent sort of the passive data collection modalities. And then we have a range of things that we want to collect, like questionnaires, we have various digital tests, for example, being able to kind of collect samples of people's voice, but essentially anything that someone has to do something actively to generate a piece of data, we have the scope for doing that. Okay. So how do we go about doing this? So we have a phone which can basically access a network, and we have various sensors and data sources, and then we have various places where we might want to actually use that data or do something with it or store it. And the thing that's sort of missing is the kind of platform that sits in the middle of this. Okay, I didn't seem to be able to go backwards, but in effect, on the last, okay, there we go. So sorry, apologies. There seems a bit of a lag with the slide changing. In effect, there are three main challenges around the platform. One is around the volume of data. So as Matthew alluded to, this is really a very different ballgame to the kind of data collection we've typically done in the past, which is kind of episodic and or reasonably infrequent. These kinds of things are like collecting accelerometer data at 32 hertz, so 32 samples every second, continuously 24/7 a day, 24 hours a day, 7 days a week, and that generates a very large quantity of data. We also have to deal with the sort of speed at which the data is arriving. So if we want to do something with that data, we've got to work out a way to kind of get it in a timely fashion to the kind of back-end infrastructure where that data will be processed. And then secondly, we've got to kind of just to deal with a kind of huge variety of the types of data that we're collecting and making use of. This is a little bit of an out-of-date slide, but gives a kind of basic flavor of the types of platforms that we have at the moment. And I've kind of ordered them a little bit with the kind of the icons on the left-hand side of the slide are in the open source arena. So these are things sort of that anyone can go and download, deploy on their on Amazon or on their servers and make use of. And the things towards the right-hand side are things you might have to approach a company to make use of in some fashion. Certainly, if you want to change any of the software, you would need sort of a company. I think a recent point in case is where the contact tracing applications wanted to make a change in how the Bluetooth sensors worked for the coronavirus application in the UK, and fell foul of Apple and Google in that respect. So RADAR-based really sits on that left-hand side. It was open sourced in the sort of beginning of 2019 as part of the RADAR CNS program of work. So anyone can make use of it, make use of the apps, and make use of the integrations we have. And in effect, what we have here are the various kind of very high-level parts of this. So the first set of things to sort of take note of are the active monitoring app, the passive monitoring app. So these are the sort of two main interfaces for the participants. The passive app will deliver all of our questionnaires and our digital tests and essentially anything. So typically, this is what the users of the platform will interface with. The passive app is actually, we decided to make two separate apps rather than one for various design reasons I won't go into here. But in effect, this allows us to collect data from wearable devices over Bluetooth. And then thirdly, we also have in RADAR CNS, we have the capability to collect data directly server to server where we don't have the scope to integrate the wearable directly into the passive app. So an example of this is Fitbit, where essentially the data goes out to the vendor, and we collect that data back through the platform again, but it doesn't go through the passive app. So that data then comes through our system. It's essentially a streaming platform. We can analyze that as it's coming through in a sort of online fashion. So this is real-time analytics, and we can then do something based on some particular event, whether that's if we detected some adverse event, we could potentially trigger a response to that, or alternatively, we can store all of that data and have that analyzed later on.
So I'm conscious of time. We're on a bit of
two more slides, I think. So finally, the active app is, as I said, where we do our questionnaire delivery. So this essentially runs through a very quick kind of registration process, and you will then receive notifications for those questionnaires. So I seem to be having some problems advancing these slides, but yeah. So in any case, it'll pop up in a second. But the notifications we provide essentially trigger people to inform people to fill out the questionnaires or perform the active tasks. And finally, there is a dashboard that essentially allows us to bring all of that information together for the people managing the study. So here we have things like the location information, the participants, where they've traveled to. We've got various pieces of information around things like sleep or activity in terms of steps, and when people responded to the questionnaires or interacted with their phones. And we have kind of values around sort of questionnaire measures. So essentially, we try and bring all that information together in one place. And I will finish on that. To pass control back.
Thanks very much, Amos. One of the key parts of this study, or these studies, is that we are asking a huge amount of participants. We have participants joining the depression study for at least 2 years. In many cases, we actually have people in these studies for longer than that. And they are being asked to give data from their smartphones. In some cases, we provide a smartphone. I'm just going to request control again. I'm still waiting for that. And it places a very significant commitment on our participants in order to take part in these studies. So one of the really critical parts of what we do is to do the patient involvement work. And this has been led by my colleague, Professor Dame Till-Wykes and Sarah Simlett, and has really been a very in-depth piece of work. I'm still trying to get control. I didn't get control. My request was not. So, Trevor, is it possible to get control? I think I've got control now. Yeah, great. Okay. I didn't realise getting control was going to be such a challenge. So we have a patient advisory board. And the patient advisory board is made up, as we might expect, of patients and representatives of disease-related charities. And they have been very active throughout. We have them at our conferences. They provide a lot of support for the programme between conferences, and have been incredibly helpful and actually becoming co-authors on many of our publications. They look at questions which are important to patients. They help us produce clear and understandable materials, and they help with facilitating engagement with the use of devices. So one example, for example, is that we had a whole range of different devices which could be used for monitoring health in the studies we were looking at. And we had an exercise with in kind of user experience of getting patients or experts to actually use and test these devices properly. It's a fairly obvious point, but if you go into a study like this thinking you know the answer, you can be sure that you don't as a researcher. You need to actually see what it's going to be like for the end user. There's also been a number of focus groups on mobile health, which have really kind of had a major impact in terms of our study designs. So, for example, what are the barriers and facilitators of taking part in a study like this? What would put someone off? We discovered, for example, that in epilepsy, people were particularly sensitive to stigma and didn't want to have a device which in any way drew attention to their condition. They didn't want people asking questions. Much better if you can actually provide devices which look like regular fitness monitors and that sort of thing. And then a lot of thought again about the sort of early barriers and facilitators to use. So the critical thing is making the technology convenient, usable, and not intrusive. There was a real trade-off between research-grade devices, which might give you absolute raw data with very fine resolution, but were clunky and had pretty terrible battery lives, versus more consumer-grade devices where the work in terms of battery life had been dealt with. So these things really matter in terms of getting it right. As I said, the study design has really been driven by this patient engagement work. I'm going to move on a bit quicker because of the challenge, and of course, online service and designing the active app has been very important to have that user involvement. So I'm going to move on now to the depression study itself, just very briefly giving a report on this. So the purpose about the depression study, I don't know if I can go back actually, but the purpose of the depression study is to look at the acceptability, usability and feasibility of RMT in major depression, to improve and refine clinical outcome measurements in depression, looking at current clinical state. So is it actually clinically useful having data on sleep from, say, a Fitbit in order to actually make some measurement of what sleep is like for someone with depression? And then the final component about looking at the multi-parametric remote measurement technologies as predictors of depressive relapse. And this is a multi-site study between London, Amsterdam and Barcelona with 2 years follow-up, 600 individuals we were aiming to get with a history of major depressive disorder. And we were asking for people we were enrolling people who had at least 2 lifetime episodes of depression, the most recent episode in the last 2 years. So we wanted people who were likely to have a further relapse, people without major medical comorbidities, by which we meant things which would mean that they had periods of time in hospital and so on. Ditto pregnancy, although some of our participants have become pregnant during the study and we've followed them, and psychiatric comorbidities as listed there. And the critical thing was that they had to be able and willing to use an Android smartphone for the duration of follow-up. So some people we enrolled were used to Apple devices. We were asking them to actually switch to an Android, which of course is quite a big deal in terms of what you do day to day. And the things we were measuring were sleep and physical activity, the active measurements from the app, speech tasks, cognitive function, mood assessments, and experience sampling methodologies, and then all these devices, all these passive streams which you can get off a smartphone. And this is the study design in a sort of in a schematic format. You have baseline to 2 years with passive and active remote measurement technology going on all the time, and then outcome measurements every 3 months, which were done on the basis of a more sort of traditional measurement of outcome based on standard rating scales. So this is our progress. I'm not going to give you much more, but to tell you that the sort of standard, the standard problem of trying to get people into a study, but actually this was not a difficult study to recruit to, and we ended up exceeding our recruitment targets of 600. And we had a lot of very enthusiastic participants. We still do. They remain very much engaged in the study. So I'm going to hand back now to Nick Cummings, who's going to talk a little about the RADAR CNS analytics.
Thanks, Matthew. Can you hear me?
We can.
Good. And just requesting control. Okay, seems to all be working. So hi everybody, and thanks for the time thanks for giving me the time to talk about the analytics this afternoon. So yeah, my role in RADAR is part of the analytics efforts. So I'm just trying to get the slide to change. Yeah, so my role in RADAR is to sort of aid the analytics effort, and we really have sort of 2 main aims within this. And the first, as you sort of heard, is there's a lot of data coming into the RADAR study. So we need to find ways to efficiently organize this data to actually be able to perform the analysis and the prediction that the clinical team like you want us to actually do. And then, of course, once we've organized the data, we actually need to perform the analysis and the prediction. And it's really currently at the moment really within this organization phase that we're sort of doing our main efforts and research. There's a lot of challenges associated with organizing this data. It is very, very multi-parametric. We have massive variations in the sort of temporal resolution of the data stream. So it takes a lot to be able to combine these things. We have the sort of passive sensors where we get data sort of every 2 milliseconds, things like accelerometers, things like GPS. We get that very, very frequently. This then sort of broadens out to sort of questionnaire data, speech data that we collect every 2 weeks. And then through the REDCap system, we're getting sort of clinical data with our main sort of predictive targets coming in once every 3 months. So we need to sort of find a way to be able to combine the power of all of these data, because there's not much point in sort of looking at one stream individually, because the real power for it and the conditions such as depression is going to come when we start to combine multiple sources of information, and especially as we then push that forward into sort of machine learning, which I'll talk about later. We also have to find ways of sort of summarizing this data over sort of different time windows. There's no point of talking about sleep if we just look at one particular 24-hour period, being midnight through to sort of midnight the next day. We need to think about how to align sleep data with activity data in ways that sort of make sense with the activities people will be doing and how they'll actually, when they're actually doing these. The other data challenge we have is that we're working with very, very real world data. So as Matthew sort of mentioned earlier, you can get very good data in things like sleep labs when people come into IOPPN and we wire them up and we get them to do all sorts of tests. We get very clean, lovely data, but in something like RADAR, it's a real different sort of ball game. Data can go missing. We can have participants forgetting to wear their devices, participants forgetting to complete questionnaires. And of course, in something like a 2-year study, we're always going to have malfunctions, things break, batteries go flat, and all of this sort of results in missing data. And just here's on this slide, you can see this little example. We can get these periods of sort of constant heart rate coming in from Fitbit data. If that was a person and that was their real heart rate, they'd be very, very unhealthy. And we can also have these periods here where we just, for no real particular reason that we can find, we can sometimes have the data go missing for a few minutes as well. And of course, part of being real world data is it's often noisy and sometimes incorrect. So a person might not be wearing their device properly. Their Fitbit might be a little bit loose on their wrist. That could explain maybe why some of the data is going missing. We ask people to record their speech data in nice, quiet environments, but of course, people still record outside when they're on the bus. This introduces unwanted noise into our system, which can help us not sort of realize the true predictive power of the different data streams. If there's just too much noise in there, we can't sort of find that signal that we're looking for associated with depression. So we really needed to come up with a sort of very huge pipeline actually to really start to organize this data. So we have our different data sources, as I've already mentioned, coming with the sort of different forms of temporal resolution. So we record different pieces of information from them. Something like the Fitbit, we might get the heart rate, the step count, sleep information. Something from the phone, we might be getting quite a lot of different variations from the GPS through the speech and the questionnaires. And as I said, through REDCap, the clinical data. We then need to check the quality of this data. Is there too much missing in a certain time period? Can we think, okay, is this reliable, good data we can actually use? Are there outliers within this data? Is someone's heart rate 300 beats a minute or something ridiculous that we know is going to be a false reading? So do we try to remove these? We also want to do some parameter extraction. So we've got a lot of information here. And what we want to do before we do the analysis is try to reduce the complexity of these, try to pick out salient, useful parts of information within this, and then push that forward. And then we sort of summarize everything. So we get the data coming in terms of milliseconds or minutes, and we look at daily summaries of these, the number of steps per day, number of per steps in the first hour of the day, the second hour of the day, things like this. We also might sort of sum up information relating to the missingness of the quality of the data, which in itself might be a marker of somebody's mental state. So look at maybe something, a percentage of missing seconds of data per hour. Then we sort of aggregate this again to try to get this in line with clinical data. So the PHQ-8, the test that the patients do bi-weekly, we might sort of do summaries based on a fortnightly period. We might then have to summarize this over a sort of 3-monthly period to really look and be able to then target our analysis directly at the sort of clinical scores and the, as Matthew said, the sort of idea of trying to predict relapse before it happens when we get these relapse measures once every 3 months. So this then, all of this sort of information feeds into our data analytic modules, where we're trying to really track the progression of the disease and monitor for relapse within this. And there's a lot of different ways we're actually going to explore to do this within RADAR. We're going to do either knowledge-driven analytics or data-driven analytics. So knowledge analytics is really hypothesis testing. So we know something about the data, we know something about the disease, we set a hypothesis, and then we use this, test this using statistical models or analytics. An example of this might be something that we know the autonomic nervous system dysfunction is associated with depression. We know this dysfunction is also associated with decreased heart rate variability. So what we're going to do here is we're going to explore relationships between a feature such as the difference between the maximum and minimum values of heart rate over an hour, and then test that and look at this against someone's likelihood of getting depression relapse and determine if there's a statistical relationship there. So very much using what we know about these different conditions and what we know we can measure in the data. And then the other end of the analytic scale, we're going to be performing the data-driven analytics. And this is really discovering new or different relationships, hopefully using machine learning. So machine learning is a very different way to sort of classical computer programming. Classical computer programming, we sort of have our data, we have a set of rules we want to process this data by, and then we sort of get an answer out from it. But in machine learning, we actually flip this around. We now say, okay, I have my data. So this is the data that we're recording from the smartwatches using the apps that Amos talks about. We have our answers. This is the clinical information coming in. And now we actually want to learn the rules. We want to know what is this relationship between the data and the answers, and learn these rules. And often these rules are highly complex and things that we might not be able to sort of comprehend through looking at the data, or things that might uncover new sort of relationships between the disease and the actual data that we've got coming in. But why do we want to do this sort of machine learning, this more complex analysis? And in one way, even though it's more complex, it's actually time saving. It's very, very difficult to write straightforward rules between certain tasks. And a very simple example here is telling the difference between a cat and a dog. As you can see on my slide, you can easily tell which one is the cat and which one is the dog. Now imagine having to start and write a computer program to actually determine this. You can't sort of say fur, face shape, things like this that we can sort of see these differences, but they're subtle, and trying to explain these into a computer system would be very, very difficult. So it actually helps sort of save us time by having the machine learn the rules. And through this, we can identify patterns in the data that might not have been obvious to us and will contain some form of useful information. And at the same time, machine learning continually improves. It hopefully gets better and better as we're going through and discovering relationships for them. And it's something that we're really excited and looking forward to within the next phase of RADAR CNS to get this going. So we really want to start converting the data that we've collected into the knowledge that we're also collecting. So what are we observing? The data coming in from the apps, and we want to know this likelihood, this relapse. So we're using the machine learning to go between these relationships. And one little example I have of this, not actually related to depression, but we've been doing some interesting machine learning work from data that we've been monitoring from our participants during the pandemic. So we've been able to observe their heart rate, and we've been able to collect information relating to the onset of symptoms from them in terms of symptoms related to COVID-19. So we've organized our heart rate such that we look at the distribution of the heart rate over 5-minute periods, and then put this into a sort of spectral representation over a 2-hour period. We then feed this into an artificial neural network, a deep learning system, to try to predict the onset of the symptom dates. And so far, our initial analysis shows that we can actually get quite a high sort of recall, doing this up to sort of 95%. But we're now going through and trying to break that and really make sure that we're predicting what we think we're predicting. We've learned that relationship that we're going to learn before we sort of push this out and publish it. But we're really excited that we've got this here, and just a little example of the sort of power that machine learning can fit into these sort of clinical scenarios and help with organizing the data. So that's the last thing I'm going to say on that, and I'll hand it back to Matthew. Thanks.
So thank you very much, Nick. I'm going to call it a day there. I had one or two other things to show a photo of the consortium and to list the participants, but we actually are well over time. And if we were on a 3-minute thesis, we'd all have failed.
So I want to invite the first presenter, that's Jonathan Rogers from UCL, so one of our invited members. Jonathan, are you there with us?
I am, yes, thank you.
Okay, so if you are ready to start, I will ask Trevor to put your slide up. Are you ready?
Yeah.
So you can start.
In 1833, the Viennese physician George Pfändler described a young woman who developed strange posturing. She gradually became more and more immobile until she ceased to move completely. These were the days before the stethoscope had been invented, and she was declared dead. Her only bit of luck was that she happened to be wearing a nice dress at the time, because after she'd been put in a coffin and buried, the gravedigger decided to open up her coffin to steal the dress. He found her alive and well. She was suffering from what we today understand as catatonia, where individuals are silent, hold strange positions, can be immobile for hours and even days, and are immortalized with photos like this one in the top left. What we wanted to do was to investigate this rather mysterious disorder with cutting-edge methods. And we used natural language processing, which is using artificial intelligence to identify symptoms in electronic health records of patients in a mental health trust. We had two aims. Firstly, we wanted to find out, was catatonia dying? Was it disappearing? So whenever the AI found something that it thought was catatonia, we validated it to make sure that it actually was. And secondly, we wanted to find out, is catatonia related to inflammation in any way? A form of brain inflammation called NMDA receptor encephalitis features catatonia in about 80% of cases. And there have also been some reports of low iron in the blood of patients with catatonia in small studies. So we found the largest number of patients reported in a study with catatonia, 1,400, and we looked at them over a 10-year period. If you look at the graph in the bottom right, you find that catatonia doesn't actually seem to be dying out in our sample. And if anything, it actually seems to be increasing, which was rather to our surprise. Even when we controlled for the size of the local population, we found that it was going up. And we have very little idea why this is. It might be the recreational drugs that are being used in the local area, but we're really not sure. In terms of inflammation, we didn't find any evidence that catatonia was associated with traditional markers of inflammation in the blood, like white blood cell count. But antibodies to this NMDA receptor, which is a marker of a very specific form of brain inflammation, were 5 times more common in patients with catatonia than in other patients with mental illnesses without catatonia. And iron, which often goes down in inflammation, was lower in the catatonic group. So conclusions, catatonia isn't going anywhere. It remains a big issue in mental health hospitals and general hospitals. And there are some early hints that a specific immune response might be involved. Thank you.
Okay, so we move to our next speaker, Paris. Are you here with us?
Yes, I'm here. Can you hear me?
Okay, yes, perfect. If you're ready, I will ask Trevor to put on your slide. Trevor, thank you. You can start.
Yeah. Diagnosis and treatments of mental health disorders are currently based on categorical structures built on patterns of syndromes and their cores, which stem from a descriptivist approach rather than etiology. Only 14 to 20% of people with psychosis will have a full recovery, with the majority having recurrent episodes and relapses. Similarly, only 25% of people with depression achieve full remission and response to pharmacological treatment. The comorbidity between psychosis and depression is exceptionally high. In schizophrenia, there is a reported prevalence of depression in around 40% of people. Conversely, psychotic symptoms occur in 14 to 19% of people diagnosed with major depressive disorder. Diagnostic heterogeneity presents challenges to existing hierarchical categorical diagnostic systems, and some symptoms have been proposed as transdiagnostic features of common underlying pathological processes. Transdiagnostic approaches suggest that disorders share common etiological and maintenance processes, and that disorder-specific treatments overlook comorbidity. Machine learning approaches could aid such challenges by disentangling complex psychopathology and providing a deeper understanding of the clinical and neurobiological profile of comorbidity by taking into account the highly complex nature of coexisting symptoms, thus potentially helping the development of new therapies for new targets. My research uses a combination of supervised and unsupervised machine learning techniques, utilizing clinical neuroimaging and neurocognitive data from the multi-site Trainees study to investigate the heterogeneity of depression and psychosis and the importance of transdiagnostic symptoms. Results so far have shown that transdiagnostic symptoms show very good ability to accurately classify categorical diagnoses in symptomatologically pure groups only, suggesting a true transdiagnostic profile in the majority of psychosis and depression patients. Traditional diagnostic groups show little neurobiological basis in terms of structural neuroimaging, highlighting the need for bottom-up neurobiological-based classification systems. Using a semi-supervised machine learning approach, models built on gray matter volume and cortical thickness data suggest the presence of distinct clusters of patients with both depression and psychosis, each clinically characterized by either a profile consisting of both positive and negative symptoms or an overall affective profile with distinct neurobiological patterns. Such biologically informed clusters could potentially aid the diagnosis and prognosis of these disorders. Indeed, in the next step of my analyses, I aim to build predictive machine learning models using these biologically informed clusters and compare the prognostic accuracy to models using traditional diagnostic structures. Ultimately, this research paves the way for precision, personalized medicine in psychiatry. Thank you for listening.
Now, our next presenter, Lucy, are you here with us?
Hi, yes, I'm here. Can you hear me?
Yes, perfect. So are you ready to start?
I am.
Okay, so Trevor, please. Thank you.
What do you think of when you hear the word cannabis? What does a cannabis user look like to you? Recently, you might have seen in the news a different sort of picture being painted. A young person who's smoking what they think is a harmless plant, only to find themselves starting to hear and believe things that can't be real. Psychotic disorders like schizophrenia might only affect about 1% of the population, but they have such a profound effect on those who do suffer that scientists have made it a priority to find out what's causing these symptoms, and cannabis is often cited as one of the prime suspects. Now, most people who have used cannabis, which is about 30% of the UK, will never get a psychotic disorder. But what we do know is that people who have a psychotic disorder are more likely to be cannabis users than those who don't. The question is, what came first, the psychosis or the cannabis? Well, one of the best ways of trying to answer that question is by looking at what people do before they develop psychosis. About 1% of the general population will develop psychosis. 1% isn't a very big number to work with. So our research team went around interviewing people across the world who are at ultra high risk of developing psychosis. What ultra high risk means is that because of their family history or some minor symptoms that they'd shown before, we could estimate that about 20% of our study population would develop a psychotic disorder. And of the 344 people who we interviewed, 65 of them did. Now we want to look at what might have tipped them over the edge. So, were participants who had used cannabis more likely to get psychosis than those who hadn't? The short answer is no. And maybe that's our final answer. But it's just whether people had ever used cannabis. And after all, you wouldn't expect someone to get cancer after smoking just 1 or 2 cigarettes. So now we want to look at how much cannabis people were actually using, which is how often they were using it and what age did they start. Now, remember that young people's brains are still growing and changing, so it might be that those who start using cannabis very young might see more of an effect than someone who used the same amount but started when they were older. Another thing we need to think about is potency. Like how whiskey has more alcohol than beer, certain kinds of cannabis will have more of a substance called THC. It's THC that gets you high. Potentially, it's the THC that also makes you psychotic. Cannabis growers are breeding their plants stronger and stronger to keep up with market demand, and this might be putting people like our study population at greater risk. Around the world, laws around cannabis are changing. The best decisions we can make when it comes to cannabis are those that reduce the overall risk of harm. First, though, we have to understand where the harm really lies. Thank you.
So now we have our next speaker. Claudia, are you here?
Yes.
Can you hear
ready?
me?
Yes, we can hear you well.
Yeah.
You're ready to start?
Yeah.
You can start now.
Vulvodynia is a condition characterized by chronic pain in the vulva without an identifiable cause. This condition can present itself in form of spontaneous vulvar pain or pain provoked by touch or intercourse. Vulvodynia affects up to 28% of women worldwide, with costs in the US estimated at $31 to $72 billion annually, making it a prevalent but underappreciated condition. Despite its prevalence and impact on mental and physical health, biomedical treatments have led to unsatisfactory improvements in this population. Therefore, a broader approach is needed in vulvodynia. My PhD aims to explore and develop an evidence-based model of vulvodynia that includes the role of psychosocial factors and to ultimately test a new individualized treatment. Since we do not know if and what psychosocial factors are most important, we conducted a systematic review and a longitudinal study. The review found that psychosocial factors are indeed related to pain, supporting a biopsychosocial approach to understanding vulvodynia. Specifically, perceived injustice, body image during intercourse, and depression were identified as potentially important treatment targets. In the longitudinal study, we examined these factors over time, and we also included the psychological flexibility model. Psychological flexibility refers to the ability to contact the present moment fully, and based on what the situation affords, changing or persisting in behavior in the service of our values. Psychological flexibility consists of three facets. Being open involves welcoming and accepting life as it is. Being aware means noticing all your experiences, and being active concerns doing the things that matter to us. So psychological flexibility may provide a useful framework to understand the experiences of women with vulvodynia, and it is the model upon which acceptance and commitment therapy is based, which is also called ACT. ACT has strong research support for chronic pain treatment, but it's never been studied and tested in vulvodynia. Through network analysis, we found that in 400 women with vulvodynia, perceived injustice, body image, and depression were key central factors. Importantly, all psychological flexibility facets were key factors, showing that ACT may be a potentially effective treatment in vulvodynia. In light of this, we will conduct a single-case individualized treatment that is based on ACT, but it will also include injustice and body image principles in women with vulvodynia. Using an app, we will also see variations of these psychosocial factors over time, in addition of looking at the effect of this new individualized treatment. Basically, it would be like individualized and precision medicine, and enabling us to see how change unfolds and also to test the new prototype. Thank you.
Okay, so that's finished. We have now our next speaker, Katrina from the Psychological Medicine Department here at King's. Katrina, are you with us?
Hi, yeah, I'm here.
Okay, ready to start?
Yeah.
Okay, please, Trevor. Thanks.
The number of people around the world living with dementia grows every year, and there are many unanswered questions about how to support people with dementia, both in terms of treating their brain disease and in optimising their well-being holistically. Healthcare database studies can help address this need. Researchers use a database called Clinical Records Interactive Search, or CRIS, which is a copy of the electronic health records from South London and Maudsley NHS Trust, with patient identifiers removed. Amongst other information, this will provide detailed assessment undertaken at the point of diagnosis of dementia. Researchers have used CRIS to look at whether anti-dementia drugs work and look at the risk factors for unplanned hospital admissions, for example. Another healthcare database is Lambeth DataNet, which allows data entered in primary care in Lambeth to be used for research. One issue in using healthcare databases is that the data is not collected for research purposes, and so may be influenced by where and how the data is collected. This may mean that there's a limited generalizability. A linkage between CRIS and Lambeth DataNet allows us to consider everyone with a GP in Lambeth, and I use this linkage to evaluate any bias in the data by comparing the dementia flags in people with a GP in Lambeth between CRIS and Lambeth DataNet. The first figure on the left shows that there are 5,239 people with dementia flagged, and just over half of these had flags in both databases, but 26% were only in CRIS in the secondary care, and 19% were only in the primary care. Figure 2 compares the presence of 7 features on a relative scale where towards the centre represents lower than average levels and around the outside is higher than average levels. There is one line for the people who are flagged in CRIS only, in pink, one line for people flagged in Lambeth Datanet only, in blue, and those who were flagged in both are purple, so that you can see a different shape based on the characteristics of people with dementia who were flagged in each database. The differences in the shape are subtle, but one thing that stands out is that Lambeth Datanet cohort, those who are in the blue, have higher levels of things that might reflect frailty, such as being in a care home or dying within 4 years. This probably reflects the fact that people with frailty in primary care are not being sent to secondary care psychiatry for diagnosis of their dementia. This should be kept in mind when evaluating results about dementia from CRIS. Thank you.
Thank you for that. So we have our next speaker, Valeria. Valeria, are you here?
Yes, I'm here.
Okay, so if you're ready, Trevor, can you please put the slide up and you can start.
Thanks. So I'm going to start by admitting to you all that I hate the month of November. I do. The days are dark, cold, and rainy, and I genuinely start feeling the pressure of Christmas shopping. So I notice changes in my mood. I start withdrawing from friends, and I sometimes find it hard to leave my bed, let alone my home. As my mood lifts, however, I become way more active. So I cook a lot more, which I enjoy doing, and I also sleep a lot better. Many of these behaviors, as you might know, are associated with symptoms of depression. And as we saw in the RADAR presentation earlier, smartphones and wearable devices can actually detect some of these behaviors through their digital sensors. So for my PhD, I am harnessing the power of digital technologies to improve outcomes in people with depression and anxiety. What I will do is track people's behavior by giving them a Fitbit and a few apps to download, which automatically collects information on sleep, physical activity, sociability, phone use, and compare that to their mood, so their responses and weekly symptom questionnaires. By studying how one changes alongside the other, I want to answer the question, can we know someone's mental state from their smartphone and wearable data? An important aspect of my project is to monitor people specifically as they go through psychological therapies. This is the part where the clinicians in the room get really excited if they think about the applications within treatment. You see, clinicians rely on people self-reporting their symptoms to diagnose and treat mental illness. But of course, people's memories are faulty. My Fitbit, however, without asking me a single question, knows if I've slept well every single night, and the GPS on my phone knows if I've basically stayed at home all week. So some exciting results for me would be that digital data strongly predicts someone's illness severity and whether they will recover. But, and just as importantly, I might find that these data collection methods aren't actually that feasible. Perhaps the data is a bit too patchy to make any conclusions. You know, people might not use or engage with the technology because they might find it stigmatizing or uncomfortable to wear. What I probably will find is that further research is needed. The idea is that if we have richer, more objective data, which no longer relies on self-report and our terrible memories, if we are better equipped to detect improvements during treatment, we will make better clinical decisions. Like in the example on the slide. Imagine my device identifying that I'm staying at home a lot more, which tends to happen when I'm feeling low. Well, it might help my therapist realize my treatment isn't yet being effective and I need a bit more social support. We use digital tools for so much already. I hope we can start using them to improve treatments, push those recovery rates up, and help us through those tough November days. Thank you.
Okay, we have our next speaker, Julia. Are you with us?
I am, yes.
Ready to start?
Yep.
So then, Trevor, if you put the slide, please. Thank you.
There are 1.35 million violent offences reported in the UK every year, and they cost £37 billion. And of course, the psychological burden of this violence is also very high. Over 60% is committed by only 1% of the male population. These men typically meet the criteria for antisocial personality disorder, which I'll be referring to as ASPD. ASPD ASPD is characterized by unlawful and illegal behavior, impulsivity, irresponsibility, deceitfulness, aggression, and a lack of remorse. Unfortunately, it's a very heterogeneous diagnosis, which means different individuals can suffer from different symptoms and still meet the threshold. Adding to the problem is that treatment options are also very limited. Some people with ASPD also meet criteria for psychopathy, but most don't. Psychopathy is characterized by interpersonal affective problems like superficial charm, grandiosity, shallow affect, pathological lying, callousness, and lack of empathy. Therefore, ASPD could potentially be subcategorized into ASPD with and without psychopathy. Neuropsychological and neurobiological evidence supports this. Those with psychopathy have severe deficits in emotion recognition and show reduced threat responses, such as a reduced heart rate, whereas those without psychopathy have heightened threat responses. The two groups also respond differently to reward and punishment, which affects their future decision-making. Individuals without psychopathy also have more generic executive function deficits. Functional MRI has shown impaired neural activity in key areas of the social brain network, and structural MRI has shown reduced gray matter volume in psychopathy. Most research to date has not directly compared those with and without psychopathy. Furthermore, research on resting state brain activity and cerebral blood flow is lacking. As mentioned, treatment options are also very limited. Interestingly, areas in the social brain network have many oxytocin receptors. Oxytocin plays a crucial role in social communication, hint the love hormone, and has been shown to improve social deficits in other disorders, so there might be some potential for ASPD. My PhD thesis will compare violent offenders with ASPD with and without psychopathy and non-offending healthy controls on various neuropsychological tests and neuroimaging measures. I hypothesize that I will find differential neuroanatomical profiles between ASPD with and without psychopathy. I also expect that oxytocin will influence, maybe even reverse, deficits in neuropsychology and cerebral blood flow. My results will impact the current diagnostic procedures for ASPD, while hopefully also informing future treatment developments. This will be done by providing more insight into the underpinnings of the disorder, which are required for the development of psychological therapies, as well as by showing the potential of oxytocin as a medical treatment. Together, this will hopefully eventually reduce the frequency and cost of violence offending. Thank you.
Okay, we have now our next presentation from Isabel. Are you around, Isabel?
Hi, yes.
Perfect. So, Trevor, if we can please get her slide, and if you're ready, you can start.
Thank you. Okay. It's usually the case in psychological research that many variables influence an outcome. Life is complex. It's also often true that the sample sizes we can obtain for experiments are small and have insufficient power to detect effects. Luckily, this is exactly the kind of scenario statistical learning methods are able to handle. The statistical learning methods we're using are essentially like regression analyses, just with an extra term added called a penalty, which can make the variables of least importance shrink away, leaving us with the really key ones. This is particularly useful when you want the instrument you created in one sample to predict a clinical outcome, say heart attack, to also precisely and accurately predict the same outcome in another population. This is becoming more and more relevant as we live in an increasingly global society where many different cultural, genetic, gender, age, and so on groups must be considered, especially as we aim to create equality and access to correct diagnosis, treatment, and outcomes in healthcare. Another common scenario in psychology is that we want to know about a trait or a characteristic a person has that can't be measured directly, for example, aggression or anxiety, or genetic liability for depression. Structural equation modelling allows us to measure these latent traits using many measurable items in a questionnaire or many genetic variants from a saliva sample, for example. Regularized SEM is a novel approach which combines the two methods created by Jacobucci and colleagues. This allows us to combine many different statistical tests into one big model analysis, which can save time and has less risk of introducing errors along the way. We're applying that method to the psychometrics and psychiatric genetics examples I briefly mentioned, after running simulations to test the method on similar fake data beforehand, which is a common practice in statistics. Our first example is a Greek adult personality inventory study of 1,700 people in total, and we've looked at aggression. Our second sample is a longitudinal birth cohort study of 1,233 women who had a singleton birth with repeated measures taken over time of psychological data from the mothers and children. We're looking at the child behaviour questionnaire items at age 3.5 and the outcome of depressive problems at age 9. Finally, our third sample will be a psychiatric genetics dataset of 1,766 depression cases and 1,745 controls, where we'll look at genetic variations potentially involved in developing depression, along with some other factors like weight and height. So far, our results suggest that regularized SEM can suggest candidate items for removal from questionnaires and perform well in comparison with more traditional methods. It can also suggest that they have differential predictive power over and above the latent factor. Thank you for listening.
Okay, so we now move to the next presentation, and that's from Rachel, joining us from Imperial College. Rachel, are you around?
Yep, I'm here.
Okay, so if we can get her slide, please, and you can start.
Did you know that as many as 1 in 6 young people self-harm? It's surprisingly common, and while some will only self-harm once or twice, for about 20% of people, it persists into adulthood, becoming more frequent and severe over time. In fact, many people describe their self-harm as addictive or like a habit. We really need a better understanding of why this 20% find it so difficult to stop, even though they may want to. This is where my research comes in, which aims to understand some of the psychological processes that could be driving people to keep self-harming, making it more difficult for them to stop. So why is it that people self-harm? Often people self-harm because it reduces negative emotions, as well as generating positive feelings like relief or calmness. Naturally, we look forward to feeling good, but this anticipation or craving is especially exaggerated in addiction, and we think this could also be the case for self-harm. Anticipation is highly motivating and often leads people to act on behaviours. Imagine you've had a difficult, tiring week at work. It's finally Friday afternoon. You start thinking about that glass of wine you'll have later, imagining how relaxed you'll feel after. By imagining this, it could make you more likely to have a drink that evening. Now, a similar process could happen in self-harm, but much, much stronger. This young person might use self-harm as a coping mechanism to gain relief from lots of difficult emotions. They might imagine the relief they would feel after doing it, which could then drive them to act on self-harming. This process of anticipating is described as the urge to self-harm. However, the psychological mechanisms underpinning this experience aren't clear. We think this anticipation could happen automatically and outside conscious awareness, just as it does in addiction. And in the lab, we're using a series of tasks, questionnaires, and interviews to investigate the effect this anticipation has on behaviour in young people who are self-harming. So what's the point of this research? Well, interventions that are currently recommended don't work for everyone, and very few, if any, are targeted to help people manage this urge. But by understanding more about these specific processes, we can translate our findings to develop better interventions in hope that we can help more young people to reduce or stop self-harming. Thank you.
Thank you. And we move now to the last presentation from this group of students who are in the middle of their PhDs. This is from Libby. Are you around? There was some echo, sorry.
Hello?
Libby, okay. So if we can please get her slide, and if you're ready, you can start.
Sexual minority young adults are more likely to experience common mental health problems than their heterosexual peers. The disparity in prevalence of mental health problems can, in part, be explained by the presence of additional life stressors related to their sexual orientation, referred to here as minority stress. Minority stress includes external events, such as perceived stigma and discrimination, but also people's responses to these events, for example, hiding their sexuality. Minority stress might directly impact mental health, but evidence also suggests that it can increase the prevalence of more general mental health risk factors, including low self-esteem. Interventions that aim to improve low self-esteem are therefore a potential way to prevent the development of mental health problems in sexual minority young adults. which is vital to decreasing mental health inequalities based on sexual orientation. My PhD thesis aimed to firstly establish that self-esteem is lower in sexual minority young adults than heterosexual, and secondly, develop a new psychological intervention to help improve self-esteem in sexual minority young people. First, we conducted a meta-analysis of research papers comparing self-esteem in a sexual minority and a heterosexual sample. And findings showed that on average, sexual minority young people had significantly lower self-esteem than their heterosexual peers. To help develop the structure and materials for the intervention, qualitative interviews were then conducted with a sample of 20 sexual minority young adults, where they were asked semi-structured questions about their self-esteem and the impact of minority stress on it. We subsequently developed a new intervention based on content from the interviews and adapted from previous cognitive behavioural therapies for low self-esteem in the general population. Intervention structure included 6 one-hour weekly individual face-to-face sessions with a therapist, and the intervention comprised 3 core sessions, shown in the centre of the diagram here, and these involved responding to self-critical thoughts with self-compassion and challenging negative beliefs about the self. Remaining sessions were then tailored to the individual and selected from optional modules centred on topics arising from the interviews, shown around the outside of the diagram here. We then set out to conduct a trial to assess the acceptability and feasibility of delivering the new intervention. 24 sexual minority 16 to 24-year-olds with low self-esteem received the intervention and completed questionnaires measuring their levels of self-esteem, anxiety and depression at pre and post intervention, and then again at 2-month follow-up. Results were really promising, so 95% of participants said they found the intervention useful and that they would recommend it to a friend experiencing similar difficulties to them. Good feasibility for a future larger scale trial with a randomised control group was demonstrated, and although preliminary, findings also showed a significant increase in self-esteem and reduction in both depression and anxiety symptoms post-intervention. And 85% of participants recovered to have self-esteem in the normal range at 2-month follow-up. Thank you.
So is Peter with us?
Yes, I'm with you. Yeah.
Lovely. So Peter, you're very welcome. This is a brief presentation. Dr. Peter Lovell is the Director of the Research Design Service London, which provides a fantastic service to support research applications. So Peter, the floor is yours.
Thank you very much, Matthew, and thanks for the introduction and for the opportunity to present today. People see my slides?
Yes, we can.
Oh, that's great. It's okay then. I'll do them from my desktop. So, just a brief 10-minute overview of the NIHR Research Design Service. We are a national network. Each region in England has its own RDS, and our role is to support those putting together research grant applications. Predominantly that's for the NIHR, although we've been given the latitude to support teams and individuals who are putting together applications for any national peer-reviewed funding stream. So that can be for medical charities and research councils as well. We're funded by the Department of Health and Social Care, so our support to research teams is completely free of charge. And all discussions that we have are treated with strict confidentiality. The slides I'm going to show today are summarising the support that the RDS London team provides, but broadly each RDS in England offers the same. So in London, the RDS brings together the 4 large academic institutions. King's Leeds, we're the coordinating centre, and we've broadly divided London into 4 patches. So institutions such as the Maudsley, which is south of the river, are supported by the team at King's College London. RDS advisors are active, applied and health or social care researchers who spend part of their time consulting for RDS. So we have a wide variety of skills. We have skills in stats and trial design, health economics, qualitative and mixed methods approaches, health psychology. We also have expertise in public involvement and research ethics. And most importantly, we have extensive knowledge of preparing successful funding proposals. Increasing the volume and proportion of high quality research grant applications is a key metric for the RDS. And our role is to provide advice and guidance on study design and health research methods to meet that aim. Our support is often much broader than that. We review team composition and suggest suitable collaborators. We can help identify suitable funding streams. It's a little bit of a minefield out there trying to choose an appropriate funding stream. We emphasise the importance of public involvement and involvement at the design stage. And we also help get the narrative right, so expressing benefits to patients and describing potential impacts. We realise that not everyone gets through first time, and we're very happy to work with you on interpreting the feedback from the funder and helping you to do a resubmission. Good research applications tell a story. They're concise yet detailed. They describe a problem or an evidence gap, and have well-defined aims and research questions, but ultimately they need to pass the judgments made by funding committee, and that's where RDS can help too. We're very lucky that we're able to observe pretty much all the NIHR funding panels, and we pass back those insights to the applicants. As part of tomorrow's training session, there'll be more information on the support on public involvement and research ethics that the RDS can provide. So I'm not going to say anything in too much detail here, just to say that we have specialist advisors in public involvement. We have online resources and workshops centred around PPI. The bursary scheme we have is available to fund pre-protocol PPI. We offer lay summary review by a team of public members. So you'll hear much more about public involvement and the support the RDS can give tomorrow. Access to our service is primarily through our website, and I'm very happy to share these slides afterwards, so you'll have the link. On the website, there's information about our events, which are all being held virtually at the moment. We have a monthly bulletin and updates via our own Twitter account. Also, there's an option to request support through our website, and I'll say a little bit more about that now. So if you put in a submission for support, first of all, you'll get an advisor, and that advisor will essentially act as your case manager. They'll give you some initial feedback on the broad design of your study, the rationale. Have you considered public involvement? Have you chosen the right funding stream? They will then triage your request and pass on to specialists within the RDS to give more detailed feedback on aspects such as sample size, economic evaluation, or potentially mixed methods approach. We also facilitate additional support from other parts of the research infrastructure. So we have very good links with the trials units, with Trust R&D and other established research networks. So we can signpost you to those resources as well. I think my last slide is probably the most important one, and that's to say we really want you to contact us as soon as you can. I mean, ideally, if you contacted the RDS 4 to 6 months ahead of a submission deadline, that'll give us plenty of time to work with you. There's no set number of reviews that we can do on applications. We're very happy to work with you iteratively. 2 to 3 months is probably a sweet spot for some good input, but anything less than a month, then we're unlikely to be able to help. So I've just included the link here again to RDS. Very happy to be contacted afterwards, or for questions to appear in the chat, and I'll deal with those. I think that's my last slide. Thank you.
Well, thank you very much, Peter. Very good advice. seek advice early, and you'll get the best out of RDS. And I'd like to thank you for the support I know you've offered many of our applications over the last few years. So really valuable. People, please do make use and think about how that links in with what you've been told about fellowship applications as well. Yeah. Great. So chat in the questions in the chat perhaps, and I'll hand back to our next chair.
So I'd like to welcome Hema Chaplin, who is with us at King's College London. Are you there, Hema?
Yes, I am. Thank you, Rena.
Are you ready?
Yep.
OK, I'll ask Trevor to put on your slide, please.
So I'm going to present my thesis about redefining and exploring refractory disease. This slide is complete chaos, which I feel summarises both myself and my PhD quite well. So first of all, what on earth does refractory mean? The dictionary defines it as stubborn or unmanageable, which some may also say describes our PhDs or colleagues at times. But in this clinical context, it essentially means not responding to treatment. There are a variety of terms for this across various mental and physical health conditions, but the broader definition seems to comprise of 3 areas, as shown here in the bottom left. This last part is currently missing in rheumatology definitions of refractory disease in inflammatory arthritis. So patients with inflammatory arthritis typically have symptoms of pain, stiffness and swelling in their joints, which is caused by inflammation, which the drugs target, but a large proportion still continue to have persistent symptoms of pain, fatigue and low mood, despite good inflammation control. No. And this group are often missed out of these treatment-resistant definitions.
Oh, yeah. Sorry, just need to
Ultimately, both groups are exhibiting continuing symptoms and reduced quality of life due to different mechanisms, but both require additional treatment in the form of non-pharmacological interventions. But first, we need to be able to identify those in need of this support through an appropriate definition. The flowchart in the top left is an overview of my PhD projects to show the process involved in generating this new definition and where I've gathered evidence from. It became clear from both the interviews and systematic review that refractory disease is multifactorial and complex. At its core, it's resistance to multiple drugs with different structures, which others had already said, but it seems to be so much more than just not responding to 3 or so drugs. The persistency of symptoms and disease activity is also fundamental, but there seems to be a whole range of these discussed, so which should be included. Finally, there are so many other contributing factors mentioned, such as comorbidities as one example, that it seems a definition cannot be considered in isolation without a broader model of refractory disease. So this is what I've developed here in the bottom right. A model of refractory disease to incorporate these wider contributing factors that can be protective or perpetuating, as seen here, with the definition of refractory disease in the middle. Using the Delphi consensus method, I asked a group of experts, including patients, rheumatologists, and wider multidisciplinary healthcare professionals, such as psychologists, physios, pharmacists, and even a social worker, to vote on the specific components they feel are important to include as part of the symptoms and impact of refractory disease. Luckily for me, they all felt the definition needed broadening out, and now it includes components regarding pain, fatigue, reduced mobility, disease-related distress, or relationship difficulties, as some examples. I'm working on the final analysis for this now, and as luck would have it, I've run out of time to tell you any more. But we're now closer to covering these important but previously missing psychosocial components and having a definition that truly reflects patients' experiences. Thank you.
So next to present is Niku Damastani. Niku, are you there?
I'm here. Hi.
Great. Are you ready?
Yep, I'm ready.
Absolutely.
Ready.
Next slide, Trevor.
Imagine you're walking home from work and eureka, you've solved that annoying problem that's been on your mind for weeks. Just as you go to write it down, a police car with blasting sirens passes by and your thoughts lost. To study brain function with functional magnetic resonance imaging, or fMRI, we ask that you do a task like solve a problem while inside an MRI scanner. We then map out parts of your brain that are active during the task. We do this by measuring the difference in signals produced by changing oxygen levels within the blood while it flows into the active brain region. We visualize the active brain regions using activity maps, shown here as red blobs on the fMRI has been an extremely useful tool for exploring differences in brain function between health and disorder. These differences could link to underlying mechanisms in the brain related to symptoms and behaviors. In fact, more than 100,000 articles have been published using this technique since it was first developed. However, fMRI scans are normally performed at the same volume as that police car siren. Can you imagine how many people cannot be scanned because of this noise? Young children may be too scared, and many patient groups can be highly sensitive to sound. Also, if we're looking at how the brain responds to a task, how can we be sure that we're not really seeing the response to the distracting loud noise? Of these hundreds of thousands of studies, how many truly represent what's going on in? This is where my project, Silent fMRI, is essential. I'm testing a new scanning method developed in collaboration with our colleagues at GE Healthcare that reduces the loud noise significantly. So rather than the police car siren blasting in your ear, it's more like hearing the soothing sounds of a refrigerator humming. Results so far have not only shown that we can certainly measure brain activity with the silent method, but we've also found indications that it might reveal more information on how the brain works. In particular, I looked at the activity maps of volunteers produced by listening to words being said during a loud and a silent scan. I found that the activity maps during silent fMRI on your right represented subtle differences, on your left in this case, represented subtle differences from the maps produced during loud fMRI on your right. Silent fMRI could be specifically unmasking the most important parts of your brain for processing language, normally hidden by the processing of the loud scanner noise. My aim during my PhD is to continue exploring the capabilities of silent fMRI to see if we can indeed find more information about how the brain works by reducing the loud scanning noise. This would enable us to study more people than ever before. After all, silence is gold.
So our next presenter is Stephanie Fincham-Campbell. Are you there?
Yes, I am.
Great. Are you ready?
Yes.
Wonderful. Please advance the slides, Trevor.
We know relationships and support keep us resilient and that they are important for our well-being and even our physical health. For example, there is evidence that having a lack of social connection is as harmful for our health as smoking. Within the current climate of the pandemic, where most of us are experiencing isolation, even more attention has been shown on the importance of our relationships. We also know that the most vulnerable and isolated people have been the most adversely affected by the pandemic. Social network interventions have been shown to have similar effectiveness on drinking to other psychosocial interventions, and they are recommended by the NHS. Outside of treatment, research has found that peer support groups can increase contact with people in recovery, providing more support for abstinence, reducing relationships with people who drink, and increasing self-efficacy in high-risk situations. I aim to summarise the ways in which social networks have been conceptualised in the literature, focusing on the measurement of social networks in alcohol dependence. I identified all of the questions from social network questionnaires that have been used in alcohol dependence. Then I organised these questions into constructs and domains to provide a new consolidated conceptual framework. I initially found 753 articles. After screening and applying my inclusion and exclusion criteria, I was left with 8 questionnaires which were used across 36 studies. I regrouped the 123 questions from the 8 questionnaires into 14 social network constructs, which were organised into 3 broader domains. Firstly, social connection, social contact, and substance use status of network were organised under structure. Secondly, general support, practical support, problem solving, social support, emotional support, confined in relationship, alcohol specific support, and being supportive were organised under function. Network quality, feeling connected, and feeling valued were organised under satisfaction. Another important finding from our review was that variations of one questionnaire, the Important People and Activities questionnaire, has been used in 77% of the studies I identified. This was the only questionnaire that had been developed within addictions, but it was developed over 30 years ago. The questionnaire was also developed without the input of people with lived experience. The findings from our review have informed my main qualitative study. In this, I'm exploring the extent to which people who are dependent on alcohol and frequently attend emergency departments discuss the aspects of social networks highlighted in my review. New insights from this qualitative work could lead to the development of a new questionnaire and also have implications for how services and interventions can support people with alcohol dependence. Thank you.
Okay, I'm very pleased to welcome Georgia Larder from the University of Manchester. Are you there, Georgia?
Yes, hi. Hello.
If you're ready, I'll ask Trevor to advance the slide.
Yes, please. On your screen, you see Vladimir Nabokov, the novelist, and what he wrote to his wife about his mental state during a flare of psoriasis. Psoriasis is a common chronic skin disease that causes inflammation, usually as red, crusty, scaly skin patches. In about 1/3 of patients, inflammation spreads beyond the skin and affects the joints as well, causing a distinctive arthritis called psoriatic arthritis. This disease tormented Nabokov so much that it drove him to the border of suicide. But do people with psoriasis have more suicidal thoughts or attempt suicide more often than other people? Although we know that psoriasis increases the risk for having depression and up to 1 in 4 people with psoriasis are depressed, we don't know if it is indeed associated with high suicidality. And we also don't know if there are specific groups of patients mostly affected by low mood. It seems that the spread of inflammation to the joints increases their depression risk, but few research has looked into that so far. Finally, we don't know why these people get depressed or suicidal. Is it because they feel ashamed of their skin? Or does psychological stress and the brain have some deeper biological connections with the skin and the inflammation caused? Since the 19th century, physicians have been debating about what links psoriasis with low mood, but we still don't know what this is. Getting some answers is important for 2 reasons. First, of course, we need to prevent mental health-related disability and death by suicide in high-risk populations. Second, we currently treat psoriasis and arthritis using medications that target the immune system, and some of these medicines have been linked to higher suicidality and depression rates, while others have been shown to improve mood. So if we have a better understanding of mood disorders in these people, we can tailor our treatment for individuals depending on their risk and needs. So what I did is first, I tried to find out if people with both psoriasis and psoriatic arthritis are more often depressed than people with skin disease only. I surveyed patients in our psoriasis clinics in Manchester using questionnaires that measure depression and suicidality. And I found that people with joint disease have higher depression rates and more severe depression than people with psoriasis only. However, they had similar rates of suicidal thoughts or suicide attempts. But regardless of whether arthritis was present, suicide attempts were 3 times more often in our patients than reported in the UK population. So why is that? I'm now trying to understand whether there are any differences in the brains of people with psoriasis compared to the brains of people without psoriasis. It is becoming more and more plausible that the skin and the brain interact biologically mainly via the immune system. This could mean that people with diseases causing inflammation, such as psoriasis, may develop changes in their brain, which can in turn be important in becoming depressed or suicidal. Nobody has looked into the brain structure of these patients before. I'm now doing that by comparing brain images and looking whether any differences relate to the inflammation within the body of these patients. Thank you.
So our next presenter, Asha Ladwa, is from the University of Exeter. Are you there, Asha?
Yes, I am.
Great. So I'll ask Trevor to advance your slide and you're ready.
Yep, thank you.
Do go ahead, Trevor.
Depression is a highly prevalent and disabling disorder which affects over 300 million people around the world. Although we have a range of effective psychological treatments for depression, it is estimated that only around 60% of individuals go on to recover. Therefore, we really need to understand how these treatments work to continue to improve their effectiveness. Research shows that some individuals experience rapid changes in depression symptoms. Much like the ups and downs on a roller coaster, depression symptoms can initially and suddenly drop, or they can quickly soar up and then plummet down between treatment sessions. Importantly, these ups and downs of depression symptoms are associated with positive outcomes at the end of treatment. But we don't know why they occur and what makes them beneficial in therapy. Therefore, the focus of my PhD is to examine whether it's something that the client or the therapist does that may instigate these fluctuations or help facilitate better outcomes at the end of treatment. The first aspect of my PhD was to determine how often they occur, because most of the research has been conducted in trial settings, and I wanted to establish if they occurred similarly in regular clinical practice. So I examined rapid drops and sudden worsening in depression in over 9,000 patients who attended NHS therapy services across the southwest of England. We found, like in trial settings, both patterns of symptoms were associated with good treatment outcomes, despite occurring at slightly lower rates in clinic settings. So, we know the rollercoaster tracks of therapy can be similar in clinical research as they are in the real world. Next, I wanted to see if I could determine why they might occur. Together with researchers at the University of Delaware in the US, we listened to over 400 therapy sessions of before, during, and after these rapid fluctuations in depression symptoms. We coded for a range of client factors such as being more hopeful, engaging in processing, as well as negative aspects such as increased avoidance and negative life events. We also looked to see whether the client and therapist relationship was positive or negative. Unfortunately, the picture was not that simple. We didn't find any of these factors were associated with these rapid changes in depression symptoms. However, we do know that having these ups and downs in therapy leads to positive outcomes for clients. So for the final aspect of my PhD, I will be looking at whether it's something that the therapist does or whether it's how the client utilizes these changes in depression symptoms that leads to lower depression and beneficial outcomes at the end of treatment. By doing so, we might be one step closer to understanding the roller coaster of therapy and how to improve treatments for depression. Thank you.
Okay, our next presenter is from King's College, and it's Maria Nettis. Are you there, Nettie?
Yes, I am.
Great, you're ready.
Yes.
Excellent, go.
Hello everyone, I am Maria Antonietta, and the story I would like to tell you today is not just about treating depression, but more about treating a person suffering from depression, like Ray, the grey man who is waving at you from the top of the slide. Ray is part of a group of 3 close friends. Unfortunately, there was a time when they all suffered from depressive symptoms. Ray in particular had very low mood, no energy, no appetite. Some days he didn't even want to get out of bed to go to work. His family was getting increasingly worried, so during one of his sleepless nights, he decided to ask for help to the doctor, and his 2 friends did the same. As you can see on the left, Ray's doctor prescribed him an antidepressant that can have several biological effects, but the main one is to increase the levels of serotonin. This is a chemical messenger in our body which helps with sleep, eating, and mood regulation, but its levels might be low in people with depression. So Ray and his 2 friends started taking antidepressants, and after a while, both Ray's friends felt much better, and they went out to the park to celebrate together. However, as you can see below, Ray was quite disappointed because the treatment did not work for him. He felt exactly the same as before. And that's where my PhD comes in. As you can see on the right, I tried to find a way to help Ray, whose depression probably was not only related to a deficit of serotonin. With a blood test, I found that he had an alteration in his immune system. This acts like an army of soldiers defending our body from stress or infections with a process called inflammation. However, Ray's blood tests showed that his immune system had much more soldiers than needed, so that inflammation in his body was higher than normal. And I thought that this could contribute to his depression. When Ray decided to take part to the MINDEP study, which stands for minocycline in depression, I added to his antidepressant another medication, minocycline, that can reduce the levels of inflammation. And after 4 weeks of treatment, I realized that minocycline was helping Ray with his depression because his symptoms had improved significantly. And I also identified the threshold number of soldiers in the immune system, or the threshold levels of inflammation, that patients like Ray should have in order to respond to minocycline. These results are still preliminary, but after taking part to this study, Ray was able to join his friends in their celebration for recovering from depression. And tomorrow, maybe, thanks to a simple blood test, we could predict who can be treated with minocycline and try to help that 1 person out of 3 who feels left behind in the battle against depression. Thank you.
Our next presenter is Ilias from the Department of Psychology here. Ilias, are you there?
Yeah, I'm here.
Great. Are you ready?
Yeah, I'm ready.
Please go for it, Trevor.
So to dive straight into it, 75% of the suicides in the UK are completed by men. In fact, men are 3.5 times more likely to die by suicide in comparison to women. So suicide is often a coping strategy when faced with mental health difficulties, is at least how it's conceptualized to men. And other strategies also include things like substance misuse, such as alcohol and drugs, as these difficulties are also more prevalent in men. One of the biggest reasons for this is that men are less likely to access support, seek mental health services, or even see mental health care in a positive light. Subsequently, this makes it extremely difficult to engage men with pre-existing services and are therefore branded as a hard-to-reach group. Usually, this is the end of the story, and there isn't much done there isn't much work done to identify what we as service providers and healthcare professionals can do to combat this. So as part of my PhD, we explored previous literature and analysed data from focus groups to work out what men need from services and what would encourage them to seek help. So the key things here are outlined in the green box, and they include a masculine narrative of help seeking that makes sense to them. So something that they can relate to. So the use of male role models and other men who have had mental health difficulties appears to work well here. Next is creating environments that protect their vulnerability. So things like confidentiality are really important to men. Thirdly is educating men about mental health symptoms, what they look like and how they present in men. Often their symptomology or their presentation can be slightly different. Fourthly is to outline services and where they can go to for support. So how they operate and what actually happens in therapy is really important. A lot of men have this notion that therapy is for talking about your feelings. Next is to use more solution-focused or active problem-solving modalities. So things like motivational interviewing and some aspects of CBT really appeal to men. Next is to use more informal approaches, so steer away from mental health labels and structured settings. These can be quite intimidating and overwhelming for men. And then lastly is to advertise mental health settings differently. So again, avoid the use of mental health labels, incorporate better incentives, and maybe offer different structures to help engage this population group. So from these recommendations, we went on to develop 3 pilot interventions that included them. The key thing to note here is that we had 2 formal interventions where male students had to pre-register and 1 informal intervention, which was positioned as more of a social drop-in. All 3 of these interventions worked well, and the participants saw them as positive, acceptable, and feasible, indicating that these recommendations are effective. Additionally, the informal intervention did significantly better at engaging male students who had high stigma of mental health, who had not sought help previously before, who had high conformity to masculine norms, and who were from a minority ethnic background, all of which are barriers to seeking help and reduce the chances of men accessing care. So in short, these show that men can be engaged in if their needs are considered, and more importantly, future research and initiatives should consider these recommendations in efforts to develop more gender-sensitive approaches to engage men. Thank you.
I'm not sure if the bar has got a bit stuck, but I think, yes, great. Let's go on now to Elliston Roberts, who's from the University of Bristol. Are you there, Elliston?
I am. Can you hear me?
We can, thank you.
Great.
Great, so I'll get Trevor to move on to your slide. Go ahead.
Okay. You remember puberty, that awful few years of oddly shaped bodies, greasy skin, feeling sad and loving and furious all at once. We know that most people experience puberty within the same normal age window, but there's a lot of variation in when people experience puberty across that window. Research has shown that experiencing puberty at a different time to your peers is linked to your risk of mental health problems, including self-harm. That's really important because self-harm is one of the strongest predictors of suicide, so knowing what factors are linked to self-harm is crucial to suicide prevention efforts. However, the question of whether it's experiencing puberty earlier or later that increases risk is still unclear, especially for boys. This might be because the timing of puberty is hard to measure. In girls, we can measure age at menarche, say, which is the age at the beginning of menstruation. However, there isn't an obvious comparable measure for boys, which means you have to ask them to self-report. Unfortunately, it turns out teenage boys are not the most reliable sources for their own level of development. The other question we're not sure about is whether any link between pubertal timing and self-harm is limited to adolescence, or whether any changes in risk persist into adulthood. This is important because if the link does persist, then early developers will continue to be at increased risk of self-harm even as they grow up. My PhD research has addressed these problems by using the Avon longitudinal study of parents and children, a large cohort study of the general population in Bristol, to see whether the timing of puberty is linked to self-harm in girls and boys. I measured pubertal timing in 2 ways: age at menarche in girls and age of the teenage growth spurt in girls and boys. This was calculated based on height measurements taken over several years by professionals at research clinics, so the boys couldn't lie about it. I analysed self-harm data collected at age 16 and 21 years so I could see whether any changes in risk continue after puberty is finished. It's critical that we can use mental health research to actually help people. But thinking about treatments, intervening to make somebody start puberty earlier or later might introduce more problems than it solves. So it might be more effective to reduce self-harm risk by changing the factors on the pathway between pubertal timing and self-harm. Therefore, as well as looking at whether there was a link between the two, I've looked at what factors might explain the association and that could be changed to improve self-harm risk. Here's what I found. Earlier timing of puberty was linked to increased self-harm risk at age 16, and I found some evidence that in female participants it was linked to higher self-harm risk by age 21 as well. I also found that some of this increase in risk was due to earlier developers being more likely to have more older friends, engage in more risky behaviours like smoking and drinking, and experience higher levels of depressive symptoms. These findings mean that we're more sure about how the timing of puberty affects self-harm risk and have a better idea of how to intervene to reduce that risk in earlier developers. By targeting those individuals with interventions to, for example, reduce risky behaviour or improve depressive symptoms, we can hopefully reduce the risk of self-harm too. Thanks.
Our next presenter is Callum Stewart. Are you there, Callum?
Hi, yep.
And you're from the Department of Biostats and Health Informatics. Are you ready?
Yep.
Okay, Trevor.
So nowadays we have problems in medicine, but we want to solve using computational models and algorithms. For example, clinical diagnoses via image recognition of various disorders, such as heart disease or cancer, studying electronic health records or monitoring diseases over time. However, as I'm sure many of you can appreciate, often medical problems are quite broad, they're like heterogeneous. So for causes of a disorder or the symptomology or somebody's behaviour to what is nominally the same thing can be quite different between groups or between different people. And so this can complicate a lot of modelling. Although you usually do have some sort of underlying pattern and greater similarity within certain subgroups or within individuals. So to make things a little more concrete, a fair amount of my PhD has been looking at people with epilepsy through wearable physiological monitors. So things like heart rate, movement, muscle activity, and that sort of thing, recorded through smartwatches or armbands. Epilepsy is characterized by the occurrence of seizures, and seizures come in all sorts of forms. So they can involve like particular types of movement. They might have autonomic changes like heart rate increases or flushing. There might be emotional or sensory experiences and so on. And they're very different between different people. What we would like to do is detect or even predict when a seizure is going to happen through the physiological data we collect. Taking the case where we simply want to detect whether a person is having a seizure at a particular point in time. So typically what you do, you have a set of data that you train a model on, and then you can use that model in the future to predict seizures. However, as I mentioned, seizures from one person to another are quite different, and that can hinder a general purpose detection model. So there is sometimes similarity within the seizures of a single person, but it's quite expensive and time consuming to record enough data from every single person that you might want to detect seizures in. Humans are very good at learning from a few examples, but typical machine learning techniques can require like thousands or even millions of examples to be able to like properly distinguish things. And like in this case, like we may only even have a single recording of a seizure for a particular person. So meta-learning is an approach to machine learning, which aims to produce models that can be quickly adapted to new tasks, with much less data than you would otherwise need. So usually you do this by training a bunch of related but slightly different tasks at the same time in a model with some shared parameters. And by seeing many examples of these similar tasks, the idea is the model will be able to quickly adapt to future instances with relatively little data. So we take all this physiological data in the epilepsy dataset and separate it per participant. We then go through each participant and train a model using a small amount, let's say a single seizure, of that person's data to contextualize. And then in the future, if we have a new patient, we would only really need to record, say, a day or so to get enough data, like a single seizure, to have a sort of model that will work for them, rather than having to record for a long period of time to get enough data for a single person. Work on the epilepsy data is ongoing, but we've tried a similar approach to detecting stress in also physiological data, where again we contextualize the model based on a small portion of data from a baseline recording. That showed some improvement over like the traditional general purpose sort of modeling.
Moving on to our last presenter of the day, last but not least, Demosthenes, are you there?
Yes, I'm here. Hello.
Hello. Are you ready?
Yes.
Excellent. Trevor, go ahead.
Meeting a friend or a neighbor and not remembering their name. Not being able to focus on what a colleague was just explaining. Constantly missing appointments or scheduling overlapping meetings. These are just a few examples exhibiting the type of cognitive difficulties many people with bipolar disorder regularly experience in their daily lives. Most importantly, these cognitive shortcomings lead to functional difficulties in areas such as work or interpersonal relationships. Impairments in these areas can have a massive toll on a person's quality of life. What's even worse is that until recently, cognitive difficulties received little clinical attention and evidence-based treatment options were very limited. Yet, over the past few years, a psychological therapy called cognitive remediation, or CRT, emerged in bipolar disorder research. Our group recently conducted a randomized controlled trial assessing a CRT approach called Circuits, which emphasizes learning of effective strategies to enhance cognition. This trial did show that Circuits does have a beneficial effect on cognition, daily life functioning, and achievement of personal goals. However, important questions remain unanswered. Who's more likely to benefit from CRT with Circuits? Does improved cognition translate into better functioning? In my PhD studies, I've been looking at these treatment mechanisms of CRT. And what I found is that CRT can be effective independent of patients' characteristics, such as their age and education, their illness duration or severity of mood symptoms. But those with greater cognitive impairments at baseline are more likely to benefit in their cognition and daily life functioning. However, this does not apply to achievement of personal goals. This means that even patients without objective cognitive difficulties can benefit from CRT in terms of attaining their own goals. What I also found is that cognitive improvement following CRT does translate to improved daily life functioning and goal attainment in the long term. This means that we can actually target cognition, not only to enhance cognitive skills themselves, but also to improve how well patients do in their jobs, their autonomy, and their relationships. These findings are important not only because they show how and for whom CRT can be effective against cognitive and functional difficulties, but also because they can bring hope to patients and their families for a better management of bipolar disorder in the near future, a future where the endpoint won't just be the improvement of a trial outcome, but the long-term recovery of patients. Thank you very much.
So that concludes the end of the 3MT presentations.
Well, welcome back, everyone. I think we should. It is 16:40 now. I'm delighted now to introduce Rachel Upthegrove and colleagues from the Psychosis Research Group at the University of Birmingham. Rachel's a professor of psychiatry at Birmingham and has been one of the leading lights of the translational research collaboration in mental health. It's been hugely helpful in terms of developing our thinking in that group. And just to say that the TRC mental health is a network of infrastructures with usually NIHR infrastructures, biomedical research centres and clinical research facilities across the country, which are all trying to develop approaches to early phase clinical trials and experimental medicine, the BRC remit, but in this area of mental health, obviously. So Rachel, you're very welcome, and I know you've got a couple of colleagues joining you who I'll let you introduce yourself.
Thank you. Trevor, I'm going to try and share my own slides so I can advance them. So I'm going to give that a go, and if it doesn't work, you can rescue me. Thanks. Brilliant. Okay. So yes, as Matthew said, thank you very much for inviting us to sort of gate crush your BRC afternoon. It's been really interesting. We've had a lot of some really interesting presentations from the PhD students, and it's really inspirational really to see how much is being done at King's. Can you put the next slide on, please, Trevor? Next slide. So what I thought I'd do is just spend a couple of minutes introducing, if you go back one, sorry. introducing Birmingham. Trevor, can you hear me? Yeah, we need to go back a slide.
I'm on the second slide, which is showing a map of Birmingham.
Ah, okay. I'm seeing the one after that, but maybe that's just me. Anyway, I'll start again. So that's it. So I was going to spend a couple of minutes just introducing Birmingham and then highlighting some of the research that we've been doing, particularly within the IMH and the Psychosis Group, talking about a few main projects. So Birmingham is north of London, and it's a big city, and you're all welcome to come and visit us when we can all travel again. A couple of facts about Birmingham: it's one of the youngest cities in Europe. For some time, we were touted as the youngest city in Europe, and it's a very, very diverse population. I think, unlike London, perhaps our diverse population is relatively stable, which is helpful for research purposes. And we also have some of the most deprived boroughs and wards in the country. So 37%, nearly 40% of the city live in poverty. It's kind of on the back of this that we've been really, pushing for a more sort of a youth mental health approach within our NHS services, and we've got the largest 0 to 25 NHS youth mental health service called Forward Thinking Birmingham. Can you advance the next slide, please? So on the back of this, that's it. A couple of years ago, we launched the Institute for Mental Health at the University of Birmingham, which is really primarily based around a youth mental health approach and a really interdisciplinary group. So those of you who were travelling on the tube a couple of years ago would have been confronted with photographs of me and Matthew, our director, on the tube. And what we're really doing is, for this, is not just promoting the launch of the institute, but really highlighting the interdisciplinarity of this, because Matthew and I were joined by Professor Lisa Bortolotti, who's a professor of ethics, and Paul Burstow, who ex-MP, who was chairing our sort of social policy group at the time, and so we have a really, really interdisciplinary focus within Birmingham. So we have a Centre for Human Brain Health, the usual sort of MRI, MEG, and computational group, but also that sort of really broad interdisciplinary group, and a really high and heavy focus on PPI and youth engagement. So our youth advisory group has been really integral in designing and driving a lot of our research agenda. Within the Psychosis Group and within the first episode Psychosis teams and back into the NHS, Birmingham has one of the largest early intervention and psychosis services in the country, and one of the longest established. We've retained our youth focus, so we go from 14 to 35, seeing a large number of first episode patients per year. Next slide, please. So just giving you a brief idea and overview of some of the projects that we're involved in within my group, particularly focusing around data science, psychoimmunology, and really trying to understand the heterogeneity of symptoms within psychosis to allow us to repurpose treatments. Next slide, please. Don't know if you can see that. Yeah. So for those of you who don't know about the Pronia project, this is a project which finished last year, led by Nikos Kritsalouros, 8 centres within Europe, and we're really sort of tasked with developing prognostic models to identify and to be able to predict the transition from clinical high risk to recent onset psychosis subjects. But within the sort of richness of the data, within Pronia, we've got a recent onset psychosis group as well as clinical high risk, and a recent onset depression group. And the total sample is nearing 2,000 patients. In Birmingham, we've really been sort of interested in and heavily involved in one of the objectives, which is modelling the risk-conferring psychiatric comorbidities within a multimodal data fashion. Next slide, please. And a number of papers are sort of like coming out now, thick and fast, from the Pronia group, ones that we've been sort of leading on, again, sort of unpicking that heterogeneity in terms of psychopathology and neuroanatomical markers, as well as risk-conferring environmental influences of childhood trauma. Next slide, please. And it was kind of really on the back of this sort of understanding and involvement in a sort of data science and machine learning approach that I was keen on using that in one of the sort of more collaborative projects we've been involved in the last couple of years, which is called PIMS, which is a project that I'm leading with Golam Kandakar in the University of Cambridge and a number of other great and goods across the country. What we're trying to really do is use a sort of a data science approach with an existing data sets to really understand where we should be targeting future anti-inflammatory agents in psychosis, trying to understand where within the heterogeneity of schizophrenia an anti-inflammatory agent might be best placed in the background of mixed evidence coming out of clinical trials. So we've known for quite some time that we need to stratify trials within schizophrenia, but how we do that has been less clear. Next slide, please, Trevor. So while we're sort of sort of working within the PIM study, it's really using machine learning and other AI techniques, so sort of unsupervised learning and clustering to identify the stage of illness, but also the symptom profile that's best suited for future anti-inflammatory trials. And then we're going to build that into an experimental medicine study of teclistamab in psychosis with a number of downstream deep immunophenotyping and mechanistic outcomes. COVID willing, this trial should start in 2021 next year. Next slide, please. But just to give you a bit of an idea about some outcomes from first sort of work packages in PIMS, this is a support vector machine learning model where we were using symptoms to try and predict elevated IL-6 as measured in the periphery out of the Benneman set. And using a very sort of basic predictive algorithm, we were able to predict elevated IL-6 with a very small number of variables. Interesting, with a reasonable area under the curve accuracy. Interesting thing for me is that depression and sort of really positive symptoms come up quite strongly in that predictive model. So this is something we're then going to be validating in the wider data set and exploring in more depth. Next slide, please. And just to say, sort of give an example of some of that deep immunophenotyping and other sort of ways of trying to model the sort of link between peripheral markers of inflammation and direct brain change within the PIMS project. My colleague Nick Barnes, who's a neuropharmacologist by background, is piloting and really pushing the way of inducing monocytes, so peripherally gathered monocytes, to behave and act like microglia-like cells. And this is something we're going to be trialing within the PIMS project before and after teclistamab. What we've been able to see so far, though, in the preliminary data is that we can develop monocytes to actually look and behave like microglia-like cells, both in terms of sort of staining, but actually then taking up myelin in different ways. Next slide, please. And then coming back to kind of really sort of bedside, straightforward trial work that we're interested in. So back from my PhD, I've been interested in this sort of idea about depression and anxiety being integral in the development of psychosis, and particularly relevant in first episode psychosis, and its importance in terms of prognosis and suicidal outcomes. Next slide. So in the ADEPT trial, which again we should be COVID willing starting recruitment in January, we are trialing a very simple design of sertraline or placebo over 6 months to see whether we can prevent depression after first episode psychosis, and see whether that will allow a number of secondary sort of objectives also to be realised in terms of functioning, quality of life, preventing relapse, etc., and preventing suicidality. You'll note on this slide that I haven't put the target number because of the prevention trial. The numbers we need to recruit to for this study are large and scary. So nearly 500 participants over the country. And I've put on this slide my Twitter handle because I'm just going to be shameless and say we're going to be recruiting for research assistants in the next few months. And if you follow me on Twitter, I'll be advertising jobs. So please do apply. I've seen some really sort of interesting work here, and if people are interested in joining us up in Birmingham, I'd be happy to poach. So sorry about that, Matthew. But next slide, please. What I'd really like to do now is just introduce Sian and Jack, or Lowry and Jack from our group. Also to say thank you to the wider group and acknowledge wider actors within the Institute of Mental Health and all of the collaborators that are involved in our projects. But to introduce Lowry, Lowry's a postdoc with me who's been background within the National Eden project, then worked on Pronia, now on PIMS, really interested in psychosis, understanding the heterogeneity and prognosis after first episode psychosis, particularly in terms of functioning. And Jack will introduce himself secondly, but Jack is a lecturer in psychology with a background in sort of neuroimaging, also getting more involved in machine learning to try and understand that heterogeneity of outcomes within youth mental health disorders. So with that, I'm going to hand over control. Again, just to say thank you guys for inviting us to talk, and hopefully we'll have some time for questions at the end. So over to Lowry and then Jack.
Thank you, Rachel. Right, so I just need to share my slides and bear with me. Okay, is that working? Can everybody see my slides?
Yep.
Yeah, great. Okay, thank you so much for inviting us to be part of your conference. And as Rachel said, it's been quite inspiring. Okay, so today I'm going to be presenting a novel modelling approach, so network modelling, and how this can be applied to understand psychopathology. And I'll give an example of how we've applied network analysis to try and understand the structure and stability of symptoms in non-remitted first episode psychosis. So just to give you a bit of background, so often individuals with psychosis, they can present with complex psychopathology, and particularly in the early stages of psychosis, we see that illness trajectories are often very heterogeneous, and symptoms may be fluid and interrelated. And although we know that early recognition and treatment can bring substantial benefit, there's still a subgroup of individuals who continue to have these poor outcomes. How can we determine the characteristics of these individuals who are potentially on a pathway to treatment resistance, and then identify suitable targets for stratified interventions? Okay, so traditionally, univariate approaches have been applied to understand psychopathology and illness outcomes, but there are limitations. So often mental illnesses are viewed as a sum of symptom intensities, where symptoms or diagnoses may be conceptualized as mutually exclusive. But this doesn't necessarily consider the collective behavior of symptoms and also the interdependency patterns of symptoms and how these might change over time to maintain psychopathology. And finally, it may not consider the influence of transdiagnostic features on outcome. If we move to a network framework of understanding mental illness, so within this framework, mental illness is viewed as a dynamical system of causally connected symptoms that considers all connections with other symptoms. And within this, we can create network models to reveal new structures within data. And finally, we can identify these key central and bridge symptoms. So these are really important because if they're present or activated within the network, they're likely to lead to this contagion effect where you see a triggering of other symptoms and widespread activation throughout the network. So these are really important to identify because they could serve as important treatment targets. And if we look at the literature to see what studies have used network modelling, and we have seen some studies within chronic schizophrenia, and they've identified symptoms such as paranoid ideation and non-positive symptoms such as apathy, avolition and depression. And also what's emerging from the literature is that the way in which the networks are structured, the symptoms are structured, might indicate vulnerability or resilience to psychopathology. So with a network that's very densely connected, so there's widespread connections, this might lead to this maintenance of psychopathology, whereas a sparser network with less connections might indicate resilience. And if we look to early psychosis, where we know interventions are likely to have their greatest impact, so there's some preliminary findings suggesting that I think in this one small study they showed the way in which the symptoms were connected and structured within the networks. This was indicative of transitioning to psychosis later on. These results are promising, but more work needs to be done. So to build on this literature, we wanted to apply a network approach to a large cohort of individuals with first episode psychosis. And we wanted to identify the network structures baseline and 12 months in those with and without a remitted status, and identify these key symptoms within the networks. So we used data from the National Eden study. So this is a UK-wide evaluation of early intervention services. So it was a large sample, over 1,000 participants with first episode psychosis who were between the ages of 16 to 35. And we used Andreessen's remission criteria to define our networks. So this was used from the PANS score. So PANS is a very commonly used assessment within psychosis. So very low scores below 3 over a 6-month period met criteria for remission. So if we look at how we estimated the networks, so this is an example of a symptom network. This was done through R, and regularized partial correlations were estimated using the graphic lasso algorithm. So you can see that the coloured circles represent symptoms, and these are referred to as nodes. So for example, here you'll see the green nodes represent positive symptoms, and the connections between the nodes, so these are essentially your partial correlations. So it represents an association between nodes or symptoms when all the other nodes are controlled for. And we wanted to identify these key symptoms, as I was saying. So in this example, you can see P2 and P4, which I think is excitement and conceptual disorganization. So they're very central in the network and have a lot of connections with other symptoms. So these are the ones to identify, because they're going to lead to the spread of activation. And similarly, we wanted to identify influential bridge symptoms. So these are the nodes that have the highest connectivity with other communities of symptoms. Hierarchical clustering was used to determine these communities of symptoms. And you can see in this example, so we've got all 7 items from the original PANS positive scale, but 1 item from the negative scale, forming a cluster of symptoms there. Okay, so we also conducted a network comparison test to compare the networks from baseline to 12 months. We applied bootstrapping to assess the accuracy and the stability of the networks. And finally, we did have quite a bit of missing data, particularly at 12 months. So we applied a Markov chain Monte Carlo simulation to input the data that predicted the probability of the missing items. Okay, so moving on to the results. So you can see here are the 3 networks that we estimated, and these are the results of the network comparison test. So you can actually see from just visualizing these graphs that the baseline and 12-month non-remitted networks appear very similar, similar in terms of the way they're structured and the amount of connections. I should say also, so the thicker the line indicates a stronger association between symptoms. But if you look to the 12-month remitted networks, there's a lot going on here. You've got your negative associations, the red lines, and the positive, the blue associations. So it looks like there's a lot of noise in this network. And indeed, when we looked at the stability coefficients, this network was coming out as quite unstable. So a bit of caution is required when we interpret this network. So when we look at the centrality estimate, so for the baseline and 12-month unremitted, sort of negative and positive items are emerging. But for the 12-month remitted, it was only observed depression that was the central symptom. But this might not be surprising because by definition, this network had low psychosis symptoms. So I should emphasise this is part of ongoing analyses, and it's made us think a bit how we define our groups in future analyses. Okay, so when we look at the bridge nodes, so again, so the baseline and the non-remitted groups are very similar, but what came out from these findings for us really is that the deficit type symptoms that are emerging, for example, stereotype thinking and conceptual disorganization. So these are perhaps quite commonly associated with individuals with enduring illness, but perhaps not so much earlier on in the illness course, and we thought that was quite an interesting find. And then the cluster analysis, so again, we're seeing similarities between the baseline and 12-month unremitted groups, and a lot of noise again in the 12-month remitted network. So just to summarise what I've talked about there, so the network structures remain unchanged, essentially for those who didn't remit, and the central and bridge symptoms included a lot of these deficit type symptoms, both in the baseline and 12-month unremitted group. And we saw distinct communities of symptoms, and depressive symptoms formed a separate community, which is interesting. So why is this important? Well, if we can identify these key symptoms within these networks, then perhaps we can identify individuals early on who are presenting with these particular symptoms, and then target these symptoms with interventions to see if these then lead to a global symptomatic improvement. But we do need to do a bit more work to understand these mechanisms of change within these structures. Which brings me on to the limitations. So even though we used large data over 2 time points, our design was cross-sectional. So we really do need a prospective study to infer greater causality. So this is the next step now, to look at these trajectories over time and see how these structures might change. We also had a lot of a high percentage of missing and imputed data, which could have led to a sample bias. And as I said previously, our 12-month remitted network was very unstable. And we wondered if this might have been a result of the data imputation, because this network in particular had a lot of missing values and a possible floor effect. And finally, there was a possible selection in the 12-month networks because the way in which we defined our outcome groups, which is based on the PANS items. So these items were also included within, or used rather, to estimate our network. So again, introducing a potential bias. So as I said, part of ongoing analyses, and this is something we need to address. So just to conclude, so from these preliminary findings, the symptom structures in early psychosis may be set early and remain unchanged in those who did not remit. So conceptualized disorganization and stereotype thinking, so these deficit symptoms, perhaps under-recognized in early psychosis and could be potential treatment targets, but replication is required. So thank you for listening. Sorry, over to Jack.
Yeah, over to Jack.
Thanks, Tommy. So thanks everybody for the opportunity to present this afternoon and to hear some of the excellent research that's being conducted. So my name is Jack Rogers. I'm based within the Institute for Mental Health at the University of Birmingham, and I'm going to present just sort of a whistle-stop tour, if you like, of some of the work we've been doing looking at neurobiological and environmental risk factors for conduct problems, or particularly conduct disorder in children and adolescents. So whilst not formally in the psychosis group, I've been collaborating quite a bit with Rachel and members of the group around thinking about prediction modelling, machine learning approaches, consistent with some of the work that's been doing as part of Pronia and as part of PIMS. The work I'm going to talk about today is part of a project I was involved with up until around 2018 called the FemNat CD project. So this was a large Europe-wide consortium project, the largest to date, investigating childhood conduct disorder. So we recruited over 2,000 young people with conduct disorder, as well as typically developing peers. So these were young people aged between 8 and 19. And the real focus was to take a sort of multidisciplinary approach to try and better understand the role of different behavioural, genetic, environmental and neurobiological risk factors associated with the development of conduct disorder. So what is conduct disorder? So as you heard before from Suzanne's excellent talk, conduct disorder is a highly heterogeneous disorder. And by heterogeneous, I mean sort of phenotypically and etiologically heterogeneous. And it's characterized by aggressive behaviors, so things such as fighting, bullying, vandalism, as well as non-aggressive behaviors, so things such as lying, truancy, and deceit and manipulation. And the important factor is that a diagnosis, if you like, of conduct disorder is made when these behaviors have a significant impact on the individual's day-to-day functioning. So this can often be at school, often leading to suspension or at worst expulsion from mainstream school. It also incurs substantial individual as well as familial and societal costs, and is predicted of other mental health problems later in life. It has a prevalence of around 3% in school-aged children, and actually, interestingly, conduct disorder is one of the most common reasons for referral to CAMS or to child and adolescent mental health services. It's also highly comorbid with other childhood psychiatric disorders, notably ADHD or attention deficit disorder, as well as more internalizing disorders, so things such as anxiety and depression, all of which contribute to its heterogeneity. More recently, diagnostic criteria have included subtypes for conduct disorder, and these include an age of onset subtype, so looking at childhood versus adolescent onset, with the childhood onset type considered to be the more severe, as well as what's called limited prosocial emotional specifiers, or what we might think of as psychopathic traits in adults. So these include things such as a lack of remorse or guilt, a callous or an emotional response, a lack of empathy, a lack of care or concern about their performance in important activities, and having quite a shallow or flat affect. And what's interesting is that a proportion of young people with conduct disorder will display these callous and unemotional traits or CU traits. And if you are interested to see an example of these sorts of callous traits, I would recommend watching We Need to Talk About Kevin, which is an excellent example of some of these sort of callous behaviours. So callous and emotional traits, or CU traits, have also formed a big part of our research, and what we often do is think about those with high versus low levels of these CU traits. And what we're particularly interested in is what factors might have contributed to why some young people do have high levels of these callous and emotional traits and why others don't. And also importantly, why some people go on to desist or remiss in their prognosis, whereas others show much more sort of lifelong or develop chronic symptoms and develop other mental health problems throughout adulthood. And so conduct disorder is actually an antecedent for antisocial personality disorder, or ASPD, which we also heard about earlier on in one of the talks. And so what's interesting is to have antisocial personality disorder as an adult, you will have presented with conduct disorder as a child, but the reverse isn't true. So not everyone with conduct disorder goes on to develop ASPD. And we want to try and better understand why that might be. And one way we can do that is to think about different environmental and dispositional risk factors for conduct disorder. So this is a model from a paper that was published last year in Nature Reviews Disease Primers, particularly on CD. But I think actually what's striking is that it could be true for lots of different psychiatric disorders. And actually these different risk factors are likely to operate or impact on young people at different times or different stages of their lifespan. So if you think about harsh and inconsistent discipline or harsh and inconsistent parenting style, then this is perhaps more likely to be important in influencing risk during childhood, whereas things like associating with deviant peers is much more likely to be important during adolescence. And indeed, that's something that we do see in terms of those that go on to have this adolescent onset type of conduct disorder, so often associating with different peer-to-peer groups around the age of 15. So this prompted some work that we did as part of the project looking at parenting style So what we did was, and this is published early this year, so this was work conducted by Dr. Ruth Pawley, who's now a postdoc in Birmingham. And what we did was to use a novel machine learning classifier and take a sort of data-driven approach to try and look at reports of parenting behaviour, so both youth report and parenting reports, as different grouping variables within our sample. So by that, what I mean is that we used a data-driven approach to try and extract key features or characteristics of parenting style that might be driving differences within our different groups, so those with conduct disorder and those without. And what we did particularly was to explore those with low levels of callous and emotional traits, or LCU, versus those with high levels of callous and emotional traits, as compared to more typically developing young people. And what was quite striking is that actually negative parenting contributed to the CD group as a whole. And negative parenting is actually quite a common feature of parenting. This inconsistent discipline and more sort of corporal punishment is quite a common feature of parenting in young people with conduct disorder. So here you can see, relative to typically developing young people, those with both high and low levels of callous and emotional traits, these features of inconsistent discipline and corporal punishment were quite striking and came out as key characteristics. When we compared just within the CD group, so comparing those with high versus low levels of callous and emotional traits, you can see that positive parenting contributed most to the module. So these were the most significant features that were extracted. And this is quite interesting because actually with those with high levels of callous and emotional traits, they tend to be quite insensitive to negative parenting, but they do respond much better to sort of interventions and positive parenting type styles. And this has been shown from adoption studies, so work from Luke Hyde in the States, and also parenting style intervention and treatments that show that positive parenting is far more effective in young people with these callous and emotional traits. So here, positive parenting includes things like more maternal involvement, supportive parenting, sort of more supervision in tasks that they enjoy. So, excuse me, the summary really then is that whilst positive parenting can distinguish within the CD group, negative parenting is much more associated with the CDUs as a whole. However, what I would say is that low levels of positive parenting is not the same as caregiver absence, and importantly, high levels of negative parenting is not the same as maltreatment. But there is important work that's being done looking at gene-by-environment interactions in conduct disorder as related to maltreatment. And that's something that we're also doing as part of our research, thinking about how gene-by-environment interactions might have an impact on things such as brain structure and brain function. So if we go back to our model of environmental and different dispositional factors for conduct disorder, then again, you might think that genetic factors are likely to exert different effects on young people with conduct disorder across different developmental stages. But some of these effects, particularly environmental risk factors, are likely to be mediated by things such as epigenetic alterations, things such as alterations at not only the genetic level, but also at a behavioural, neurocognitive level, and a neurobiological level, as related to things such as brain structure and brain function. And these differences in brain structure is something that also we've been exploring. So a few years ago now, we published a meta-analysis, the largest meta-analysis to date on this topic, to really provide some more coherence in the sorts of differences in brain structure, in this case grey matter volume, you might expect to see in young people with significant conduct problems. So we used 13 studies that adopted a technique called voxel-based morphometry, and we obtained raw data from a lot of these studies. And what we did was to look at differences in grey matter volume between young people with and without conduct problems. And what we found is there was significantly reduced grey matter volume within a number of key regions in the brain that have been implicated in things such as emotional regulation, things such as face processing, executive functioning. And these included areas such as the amygdala, the insula, and the left dorsolateral prefrontal cortex. We also have some more recent work from last year published using, not looking at this time at grey matter volume, but rather at white matter differences. So white matter pathways are the sort of structural highways of the brain, if you like. So they're the pathways that connect the grey matter regions together. And sadly, I don't have time to talk about that today. But what we did find is, again, structural differences in the brain in these white matter pathways that connect some of the grey matter areas that have been shown to show reduced volume in young people with conduct disorder. What we also found is that there was a mediating relationship between structural changes and other personality measures such as CU traits or callous and emotional traits, as well as performance on neurocognitive tasks such as measures of impulsivity. So in our case, a go, no-go task. And these different features and thinking about them in unison, I think is quite nice and very important. And so what we've been doing more recently is thinking about how we could maybe classify and take a more data-driven approach, classification of conduct disorder using machine learning techniques, so sort of semi-supervised approaches. So we know that some of these prediction models trained on both clinical and neuroimaging data can help identify features that inform things such as comorbid symptoms, things such as recent onset and risk for long-term prognosis, particularly in work that's been done by Rachel and other people around psychosis, and work being done by Paris, who you heard from before, particularly around recent onset psychosis and depression. And there has been some work done on things such as child psychiatric disorders, such as ADHD. But interestingly, nothing has really been done looking at conduct disorder. And this is perhaps surprising given the heterogeneous nature of the disorder and obviously the high rates of comorbidity with other childhood psychiatric conditions such as ADHD and depression. So what we've done is, and this I should say is still quite exploratory, so work that we're working on now. So what we did was to train two separate semi-supervised linear support vector machine models using the Pronia sort of neurominer pipeline, which utilizes voxel-based morphometry type techniques for segmentation of the brain. And what we did was to try and classify CD used from typically developing young people in two separate models. So the first model adopted more clinical features. So these included socio-demographic variables such as age, gender, pubertal scale, as well as self-assessment and personality measures such as those like CU traits, parenting style, measures of childhood trauma and maltreatment, as well as empathy, and comorbid symptom profiles as measured during our clinical assessment that we did with all of the young people and their parents. We also had a model that focused on the structural scans, so looking at grey matter differences and segmented grey matter areas of the brain of all these young people. And what we found, interestingly, is that the clinical model outperformed the neuroimaging model, and there are a number of reasons for that, but the clinical model was able to classify the conduct disorder participants with around 90% accuracy. So if we just unpack the image you're seeing here, the blue dots are instances when the model correctly classified conduct disorder young people as having conduct disorder, and the red as classifying those typically developing young people, and the stars represent the instances of misclassification. And so I think what was quite striking as well is that the key features that came out from this clinical model included things such as age and gender. So I should say that the FEMNAT CD study was particularly focused on looking at differences between boys and girls, with a lot of the work to date focusing only on boys with conduct disorder. We also found a relationship with comorbid symptoms, particularly those that were commonly comorbid with conduct disorder, so those such as ADHD and oppositional defiant disorder, or ODD, as well as measures of depression. And interestingly, what came out, which is perhaps which was reassuring in some ways, is that features associated with manipulation, with grandiosity, with lying, manipulation, these sorts of callous traits also came out within the top sort of 10 features that were particularly important for classifying within the clinical model. To give a bit more information about the neuroimaging model, it did reveal, interestingly, reduced grey matter volume within a number of areas that actually are consistent with our meta-analysis that I talked about previously, which again is quite striking and for us quite reassuring. So these included areas such as the insula, which also survives whole brain correction, areas such as the insula and the dorsolateral prefrontal cortex. So areas implicated in particular processes that have been shown to be impaired in young people with conduct disorder, such as emotional processing, decision-making, impulsivity, aversive conditioning. So the next step is really to see how the model performs when we combine both the clinical and the neuroanatomical models together, and that's something that we're currently doing now and finalising. So just to summarise then, so hopefully you're convinced that chronic disorder is a highly heterogeneous disorder. It presents with an increased risk for mental health problems across the lifespan, but actually despite that it remains a very understudied disorder, and that's despite the fact that it's one of the most common reasons, as I said, for referral to CAMHS. It's important, at least we think, to incorporate different data sets and take a much more multidisciplinary approach to a better understanding the environmental and neurobiological components or risk factors associated with chronic disorder. And this includes thinking about different profiles, gene-environment interactions that can help better explain developmental etiologies in the disorder. And also, most chronic or severe trajectories of disruptive behaviour begin very early on, and this is shown not only in those young people that present with these symptoms very early, but that actually early intervention is key in terms of preventing mental health problems throughout adolescence and into adulthood. And so we've recently been reviewing some work into multi-systemic approaches and different social learning principles, and what we're really interested in is whether some targeted interventions could be better informed by more novel analytical approaches such as prediction modelling or machine learning type techniques that I've discussed. The hope is also to try and get a better understanding of the underlying pathophysiology that again we can use in the context of subgrouping and these callous and emotional traits. And what we're really excited by is obviously thinking about interdisciplinary collaboration, so work we're doing with the Psychosis Group around maybe inflammatory markers and association with grey matter change, more prospective or longitudinal studies, and the importance of sharing these sort of large data sets. So we're currently part of a working group in the Enigma project, which has a lot of data looking at meta-analyses through neuroimaging and genetic data. So I'll stop there and say thanks ever so much for listening.
Great. So Matthew, I don't know whether we have any time for questions. We probably don't because we've gone up a bit over time. But people can follow them up, or we can. It's a bit hard on the format anyway.
I suspect not. I'm really sorry because it's really fantastic, rich presentations, and thank you so much.
Yeah.
Larry, Jack, and Rachel for coming and joining us, and it's wonderful to hear about the range of work going on. And we'd be, I mean, I think the point about the TRC mental health is to foster collaboration. The point about this conference was to sort of dip our toe into having a bit more, slightly more permeable boundaries of our work and to showcase some of what we're doing. But hear from a number of centres. We've had, I think, 7 presentations from of the 3-minute thesis presentations and yourselves. So thank you so much for joining. And I'm sorry we're not in an auditorium when we could have that all too valuable chat afterwards and catch up, which I think we miss desperately now. But I think we probably should move on if you don't mind. Just say thank you very much for inviting us, and when we are back in a real world, we'll come down and have the chat and the drinks afterwards, definitely. Well, welcome to Birmingham. Yes, even better. Great. Okay, well, it now falls on me for the closing remarks, and I was hoping that we'd be able to announce the winners of the 3-minute thesis competition. I think we can for the audience winner. Is that right, Julia?
Yes. We're going to announce an audience winner for each session and also the overall audience winner, but there's not actually a prize attached with the audience voting. Ah, but the prizes come from the judged ones, and we had an issue. Yeah. The issue is that we're not used to the voting system, and I certainly got caught out by this. That if you didn't hit the return key, your vote didn't get registered. And the problem is that if you didn't hit it within the time you had, the thing just went blank. So we didn't actually capture everything. So Julia's now getting everyone else. I think I've got all mine in, but everyone else who was a judge to do their bit. And we want to do that properly rather than sort of wing it now. Yes.
So it'll be announced tomorrow on, how will it be announced, Julia?
I should be able to email everybody, but if not, we'll put it up on the website. So we'll put it up publicly who the winners are. I should also just add that the judging scoring was slightly more complicated than the audience vote. So the audience were entering just one score, whereas the judges were entering 2 scores plus feedback where they were able to. So that's why it's been slightly more tricky with the judges scoring.
Yeah, and the judges are also compromised by being people like me. Okay, so apologies for that.
Julia, a quick question. The scoring that the judges were doing it on the basis, are you happy to share what the criteria were?
Yes, the criteria are the 3 MT. So we'll actually put that up on the website. So when we announce the winners, we can put the judging criteria up. So it's the official University of Queensland 3 MT competition criteria.
So I want to now just close, and I guess I'm gonna announce when I get them. I'm going to announce the results of the audience prize.
Sending them to you now, Matthew.
Sending it to an email now. Well, I'm going to leave that there for the moment. I had a fantastic afternoon. It was such a refreshing afternoon compared to most of the afternoons I have of endless meetings about COVID and how we do this, that, and the other, and it was, you know, even though we're on Teams yet again, I thought the format of the 3-minute presentations worked incredibly well. Sorry, I'll stop that. And thank you everyone who's participated, particularly those of you who did the presentations in the 3-minute thesis. I don't think anyone went over time. It ran beautifully smoothly. Those components stayed perfectly to time. It was the more senior academics who went over time in the 2 research showcases, but it was a fantastic achievement, and I think it's also quite difficult to find clear winners here, because I thought the standard overall of people's presentations, the different ways in which people presented, was really marvelous. But I want to do some thanks, and I want to send particular thanks to Trevor and Hannah, who've supported the planning and running of the AV. I'd like to thank Saleha, Serena, Alex, and Chanpreet, who helped with the planning and particularly the comms around this. To thank the chairs of the different sessions, Richard, Rena, and Patricia, who also helped in the planning and bringing it together. To thank Deon and Claudia, who are both first-year PhD students, for organising the social event, which is going to happen in a moment. To thank all the presenters, I've done that already, and to thank the workshop presentations this morning. So I was able to join the first workshop, which Niku put together, and was a really wonderful occasion. Actually, it was great just talking about race in research and how it works, but also to Patricia, Peter, Sam, and Stuart, who were involved in the in organising the second workshop. Niku, Ben, Ilias, and Gemma, who helped on the BRC representatives since 2018 and are now stepping down to concentrate on their thesis writing up. Those roles are incredibly important. It really helps, make sure that the training program we run, that there's sort of good communication operating. And also, I think to say goodbye to a number of our final year students who started in October 2017 at the beginning of the current BRC, and to wish them every success the next steps going along in their career pathways. Now, I've got a couple more thanks to give, but I just want to check in on the email, which I hope I've received. You're going to watch me fail to open up because exactly that. I'm all fingers and thumbs. I'll get on this one. Here we go. The tension's unbearable. So, scrolling up, audience winners. So, So, the first session, Timothy Lawn. Second session, Valeria de Angel. And the third session, Ilyas Saga-Rigali. I'm sorry, I've probably massacred your surname. And to say the overall winner, Valeria de Angel. Valeria, Val, many congratulations. This is absolutely nothing to do with the fact that Val is my own, I supervise Val. I'm absolutely delighted for you. I think you did a fantastic job there. So many congratulations to those winners, and we will celebrate. We need to find a way of celebrating these things better. But many, many congratulations, and I can see a whole load of congratulations going up on the chat. Salah?
I was just going to say that I'm sure you'll probably add it right at the end, so my apologies for preempting you, but the conference would not have happened without Julia. So
thank you, Salah. I was about to say that.
Sorry, I'm so sorry to take away from that.
And I wanted to say two very special thank yous. One to Julia, who runs these programs, these conferences, and actually the whole education and training program, the training and capacity development program, with incredible kind of calmness, professionalism, good humour. She tolerates people like me messing up the voting system or running over time with complete equanimity, and does a wonderful job. And I think everyone here, if this was the auditorium, it would be absolutely ringing with applause to thank Julia for what she's done. So thank you very much. And the second person I wanted to thank is Richard Brown. Richard mentioned that he was stepping down in a couple of weeks' time. Richard is far too young to retire, but he is retiring, and he has been with the BRC as the training and capacity development lead right from the get-go. Richard is modest, so he won't like me saying too much, but I'll say a little bit. And what I want to say is that with things like the BRC, when you're organising competitions and infrastructure, things like a training and capacity program, you really want to know that it's kind of being done in the right way, that proper attention paid to all the things we expect with competitions, that are operating fairly, without fear or favour, and everyone can feel confident. And if you're in my position as a BRC director and something isn't being done right, my God, you get to hear about it. So the fact that I never get to hear about any complaints about the way in which the system has operated is absolute endorsement of Richard's leadership. And he's just been an incredibly supportive, calm presence, whose input I have valued enormously, and I'm going to miss Rich hugely. I'm very glad that Rena has agreed to take on the role, and I'm absolutely confident Rena's going to be able to do a wonderful job. But I would really like to everyone to join in applauding Richard. And I don't know if Richard would be, we could twist Richard's arm to say a word, but Richard, many congratulations and thank you.
Yeah, coming out of the shadows. Good. There's some gifs and things flowing around in the chat, and a picture of me blushing, my little emoticon. Actually, I look more and more like the emoticon every day since I had my last haircut. So, well, I just want to thank you, Matthew, for the kind words. Very generous and very embarrassing. I'm very appreciative that, well, very thankful that the work which I and people I've worked with have put in over the years has been A, recognised, but B, as well, I think, produced such an outstanding crop of early career researchers in terms of the numbers that have gone on to stay not just here with us at King's, but also to go elsewhere around the UK and internationally as well. So I think we've more than met our remit from NIHR in terms of greatly increasing research capacity in the area of mental health, and will continue to do so in the coming years, all being well, and future funding. And I'm more than happy for Rena to pick up the baton at this point, and I hope Julia will be there alongside her to make sure things go from strength to strength. So thank you, Matthew, thank you, Julia, thank you, Rena, thank you, everyone. Too many people to mention. I very much hope I'll have a chance to have a sit-down face-to-face meeting and possibly a small drink at some point in the not too distant future. So thank you and goodbye. Crikey, that sounds funny to say, but so long.
So long, and it'll be very good quality gin, I'm sure. Thank you, Richard.
Thanks, Matthew.
The final pointer, the evaluation online. You can see it on your screens now. Please do it. It really does actually make a difference. We do look at the evaluations. It makes a difference in how we organise these things in the future, and we'd really appreciate everyone just spending a minute or two filling that out.